I am Dr. Sushri Mohan, and I am your instructor for this class, Anatomy and Physiology Part 2. And typically we'll meet on Mondays, 10 a.m. to 12:50 p.m. We'll just go over the specific course policies. You are already aware of it by now, and then we'll get started. By that time, I think your friends would have joined. So this is my official email address, smohan@gwinnettech.edu. So if you have any emails or any concerns, you can always send me an email, and I will try to respond to you at the earliest possible time. About the course, please be acquainted with this course and that it's a blended lecture. So lectures would be online and the exams would be at the ANF campus. And if you have received a copy of the syllabus, you can go over the classes and what we are going to study in this class. Be acquainted with the schedule, grading scale, and bonus points. So our meetings are synchronous classes, online, face-to-face. Monday is 10 a.m. to 12:50 noon. And the other thing is that the meetings would be recorded by me, but I was instructed in the meeting that I'm not supposed to upload the recorded meetings. So you will not get a copy of the recorded lectures. So I would request you to attend all the classes and not miss anything. And if you are going to be absent in any specific class, do send me an email in advance and let me know. Tests and final exams are on the campus at the Alpharetta ANF campus, and homework is due online. I was also told that as per the policy change, there would not be any more retakes in exams. So if anybody misses an exams, it would be a 0. Only use the GTC email address, and you are aware of the discussion boards that you have to complete all your assignments. Grading scale. Bonus points. Well, if you attend the classes regularly and if you have attendance in all the classes, then you earn 1 point each day leading up to the test and max up to 5 points per test. The points do not roll over. So prepare, organize, work, evaluate, and review. So these are some of the basic things that we have to make you aware of. If you have any questions, always feel free to send me an email. So we'll begin with the digestive system. Most of you already, you are aware or you know that the digestive system, it starts at the school level, at the basic foundational level. So all of you have some, more or less, some idea about the digestive system. Now about ANP2, I would like to explain to you certain things. I want to make it very clear to you right from the beginning. ANP2 course is easy and it is doable, and you can certainly get good points and good grades. It is not something beyond you. You are capable of, 100% capable of passing this class and getting very good grades. The only challenge would be that these classes move quite fast. They move very fast. So a lot of information that you have to assimilate, understand, and process. And if you are not going back to the lessons and if you are not going over it, then the next class becomes a bit more difficult. And then the next class becomes even more difficult. And it comes to a point where you feel like, I can't do this anymore because it's too much of information and it's confusing me. So what I would suggest is, today is the first chapter and the first class. I would request you to go back and go over it tonight and go over it tomorrow, so that when we meet next Monday, you are already thorough with digestion. I do not expect that we have 155 slides to cover, okay, between 10 a.m. to 12:15 p.m. I do not expect you to remember all the points. But what you can do is sit patiently and watch me as I explain. I will also ask you questions so we can have time for interaction. And if you have any questions, feel free to ask me or send me an email. I have had students sending me email at 2 o'clock in the night, 3 o'clock in the night, and I don't hesitate to, reply them first thing in the morning. So if you get stuck somewhere, if you have additional questions, feel free to ask or send me an email. But move fast, because the class will move pretty fast. And it's a lot of information to remember. And I would also suggest draw diagrams. As you study, let your hands move. Draw diagrams, draw graphs, schematic representation. Be a visual learner. And as you draw and you label them, if there are functions, if there are biochemical equations, write them along. Maintain a diary. Just don't read from the laptop, but use a note copy and pen to draw, to scribble, and because as you draw, you remember the spelling as well. So this is my advice to you. And if you have any questions, feel free to ask me. Do you have any questions? You can send me your chat. I can read them over here. And if I miss any chats, you can always send me an email. So let's get started with the digestive system. Any questions? Okay, so I will begin with the digestive system. Well, the digestive system, it consists of the alimentary canal and the accessory organs. So what is the alimentary canal and what are the accessory organs? So as you can see, this is my phone. Okay, this is my phone. So this you may consider as the main device, and I use it for texting, for receiving calls, for video calls, whatever. So that's the main device, and all the information flows through this mobile phone. The accessory parts of a mobile phone are the adapter, the charger. These would be used to charge the mobile phone. Or maybe if I use earphones or something, AirPods, they would be connected. So the accessory parts are the helping parts, like an adapter to the mobile phone. The main one is the mobile phone. The same way, the alimentary canal, that's the main one through which the food or the nutrients pass through it. They pass through it, and the food is broken down and the energy is absorbed, or the nutrients are absorbed. And the accessory organs these are the helpers. They are the helpers that provide help to the main alimentary canal. So the alimentary canal, we also call it as the gastrointestinal tract. So gastro and intestinal. Why is it called gastro? The word stomach, okay, it's actually called gaster. So that's why from the term gaster we get gastro, and as you know, intestine is intestinal. So gastrointestinal. So from the word gaster, you have heard gastric juice, gastric hormones, and gastritis. Okay, that's the disease condition. So all of these are derived from the word gaster. Gaster, stomach is called as gaster. It's also called as the alimentary canal, is also called as the GI tract. That means gastrointestinal tract. But the food actually goes through it. The food actually goes through it. And it's related to the nourishment or the nutrition, furnishing the sustenance or maintenance. So digestion and absorption of food happens in the alimentary canal. Does it happen in the accessory organs? No, it doesn't. It only happens in the GI tract, the digestion and absorption of food. It happens in the GI tract. Now let's look at the accessory digestive organs, or the helpers who help in the process of digestion. Teeth, tongue, gallbladder, digestive glands, salivary glands, liver, pancreas, all of these, they play a role in digestion, a helping role in digestion. Food does not pass through them, like the GI tract, food does not pass through them, but they help the digestive system. Like information does not pass through the mobile phone, but the adapters, they help charge and keep the mobile phone alive. So this is the general pathway, the main pathway, how the food enters and goes through the digestive tract or the alimentary canal. It enters through the mouth, that is the oral cavity. Okay, and this is accessory. These glands are all accessory or helpers. Then the food goes down in through the pharynx, number two, that goes through the pharynx. Then it moves down through the esophagus, that is number three, and then it goes right here into the stomach, that is number four. Now, in the meantime, pancreas and liver and gallbladder, these are all accessory helpers. Then number four is stomach. From the stomach, it comes down to the small intestine, that is number five, and then to the large intestine, number six, and finally out through the anus, that is waste. So food enters through this pathway: mouth, pharynx, esophagus, stomach, small intestine, large intestine, and out through the anus as waste. What are the digestive functions? Ingestion, that is eating, taking in the food. Deglutition, that is swallowing. So this is a new term, deglutition, you have to remember. Deglutition is swallowing, the act of swallowing the food. You know, little kids, you give them food, they'll keep it and in their mouth and they'll hold the food in the mouth. Come on, swallow it, swallow it. We're getting late for school. So you have to push them to swallow it. Keep talking to them, asking them to swallow the food. Digestion. Now, digestion is two types. Either it is mechanical or it's chemical. I mean, it's both, but we'll talk about what is mechanical and what is chemical. Mechanical digestion is different from chemical digestion. Okay. I'll go to that in detail when we go to that slide, but for now, you know this much. Mechanical, there are two types of digestion: mechanical and chemical. In simple terms, I would say mechanical would be like a physical process of breaking down big pieces into little pieces. Okay. And who can do this job for us? Teeth, mastication or teeth, chewing, that can break the big pieces into small pieces, and that's called mechanical digestion. Chemical is when we involve enzymes, or chemicals, and that would break down the complex substances into simple substances. This is big chunk becoming small chunk. This is complex food items becoming simple, and this uses enzymes. Next work is absorption. Once you break down the food into digestible small parts, then it has to be absorbed or assimilated. The biological term would be assimilation, absorption into the blood so that you can get nutrients. And then the waste is removed by defecation. The waste comes out through the anus, the process of defecation. So ingestion, swallowing, digestion, absorption, take the nutrients out of it, and then the waste is removed by defecation. So here it shows the basic functions. First, we begin with ingestion. The food enters the mouth, and then mechanical digestion, which I just explained, which is chewing. Okay, chewing in the mouth, that is also called as mastication. Then the food passes through the pharynx. We are not going to call it as food at this point. We are going to call it as bolus or food bolus, because food mixed with the saliva at this point will be called as a food bolus. A bolus is nothing but a ball of food. It's like with mucus, it's messy, it's like a completely, it looks yucky, and it's a food bolus that passes through the pharynx, and it through deglutition or swallowing process passes through the esophagus, that is propulsion to move it forward, swallowing, and then it undergoes a motion which is called peristalsis. We'll go through all of these in details. Peristalsis is basically a wave-like motion that propels the food down. Like, it's see the wavy motion. It's like a toothpaste. Like a toothpaste, when you squeeze the toothpaste and the paste comes out like this, that's the motion or the movement of the smooth muscle of the wall of the esophagus, and all that is called peristaltic wave-like motion. So the food moves down and then it comes to the stomach. Now at the stomach, you can see the blue arrows are pointing like this. It's like a churning circular motion, mixing of all the enzymes with the food. Then it goes down through the small intestine, large intestine, and then defecation is the waste out. So ingestion, the actual act of taking the food in. Mastication would be the chewing. What we chew, when we take the food, we chew well in the mouth. That's mastication. Now here, this is important. I would want you to remember these things. Which muscles, skeletal muscles, are involved in mastication? This is a question for you. Masseter muscle, temporalis muscle. So now in the exam, I would like to ask you, which type of muscle is involved in the process of mastication? Is it a smooth muscle or is it a skeletal muscle? It is a skeletal muscle, because like kids choose when they want to chew the food and when they want to hold the food in their mouth. You feed them broccoli, they will sit with the broccoli in their mouth for 30 minutes. You feed them ice cream or any other delicious items, a KFC chicken or something, they're going to chew it and finish it. So that means it is under the voluntary control of the child. Our voluntary control, what we want to eat and what we don't want to eat. So that's why skeletal muscles are voluntary muscles. You can use them when you like, and these muscles are masseter and the temporalis muscle. Look at their position, the masseter and the temporalis muscle. So this is a question for you. Which muscles are involved in mastication? Which nerve provides the muscles the sensation or the impulse to contract? That would be cranial nerve, fifth cranial nerve, trigeminal. Okay, trigeminal nerve, remember, that provides the impulse to the muscles to contract so that you can have the act of mastication or chewing. The tongue, and they also help in closing the mouth. Now the tongue, it helps in manipulation of the food. How? Because you can move your tongue, so you can move the food bolus inside your mouth, mixing it with the saliva. And the saliva has the enzymes, the salivary amylase enzyme. So mixing of the food with the saliva. Then the teeth, they help in grinding, chewing, our incisor teeth. And if you're eating any non-veg food items, meat, then canine teeth, these help in tearing the food and grinding and mixing, break the food down into smaller particles. So these are the basic processes in digestion. Deglutition is the swallowing, and controlled in the brain stem, medulla oblongata, and pons varolii. So this point is also important. Okay, so if you have ever noticed, if a person has a brain surgery, they just went through a brain surgery, then you will find that they have difficulty, when they recover, they have difficulty in swallowing the food. Why? Because our swallowing center is located in the brain stem, medulla oblongata, and pons varolii. So that's why somebody having a brain tumor or going through some brain issues or got into an accident or something, when they're recovering, swallowing may be an issue. Swallowing is voluntary and involves skeletal muscles, as I told you. And then the skeletal muscles for chewing and also for swallowing. Muscles that are innervated by 5th, 7th, 9th, 10th, 12th. Okay, so trigeminal, that is 5th. It's both sensory and motor. That means it's both afferent and efferent. Facial, 7th, that is sensory and motor. Glossopharyngeal, 9th, sensory and motor. That means both afferent, takes the information that is afferent and brings the information back, that is efferent or motor. Salivation, so it causes it causes salivation. Vagus, which is the most important one, 10th, it's a mixed nerve, and hypoglossal nerve, that is 12th, motor. So all of these muscles are provided impulses by these particular cranial nerves. So you have to remember the names of these nerves that play a role in controlling the muscles that are involved in chewing and deglutition, which is swallowing. Any questions? You can unmute and ask, or you can just send the chat if you have any questions, or we will move on. Okay, so if you do not have any questions, let's move to the next slide. If you want me to, sorry, if you want me to repeat a particular slide which you did not understand, feel free to raise your hand or chat and tell me to please go back and repeat it. Okay, so I don't want you to move forward with any doubts or any questions on your mind. We go to slide number 19. Peritoneal cavity. So peritoneal cavity, this is not something very complicated, but a lot of the questions in the exam usually comes from this particular aspect. Like not a lot of questions, but you can always expect some questions to be from the peritoneal cavity. So the peritoneal cavity, that's the space within the abdomen that contains the stomach, liver, spleen, intestine. It is comprising of visceral peritoneum and parietal peritoneum. So now, let me explain to you. This is the abdomen, okay? And we have so many organs inside the abdomen. Let's consider that these organs are surrounded by the peritoneum or a peritoneal cavity, whereas the remaining organs are outside the peritoneal cavity. These are outside and these are within a peritoneal cavity. So we'll call these organs as intraperitoneal, and the ones that are outside, we'll call them as retroperitoneal. That's the simplest way how I would explain to you. If you are covered by a peritoneum within the peritoneal cavity, I would call you the intraperitoneal organs, and if you are outside it, I would call you as retro. Retro is containing organs that usually do not move a lot. They are mostly fixed organs. They don't move so much. And they are attached to the abdominal wall. They're attached to the abdominal wall by adventitia or fibrous connective tissue. They're attached to the abdominal wall by fibrous connective tissue, which we call as adventitia. Adventitia. Okay, so that these are retroperitoneal organs outside the, number one, they're outside the peritoneum. They're mostly fixed organs. That's number two. Number three, they're attached to the abdominal wall by this adventitia, which is fibrous connective tissue. Now look at these organs that are within the peritoneum. We call, and one is retro. These ones are called intraperitoneal, within the peritoneum. These are the organs that kind of little bit are suspended and they move around a bit. So that's the basic difference between intraperitoneal and retroperitoneal. What makes it complicated is that we have some organs that are partially intra and partially retro. That is why, for example, in my body, there are parts of my body that that are exposed to the sun, and there are parts of my body that are not exposed to the sun. So that way you have to remember that there would be organs that are intraperitoneal partially and partially retroperitoneal. So that means you have to be very specific in your answer who are intra and who are retro, because they could ask you, what is duodenum? of the small intestine? Is it intra or is it retro? Well, part of duodenum is intra and part of it is retro. So you have to be specific for each organ. Okay. So let us look at serous membranes of the peritoneum. Let's focus on the peritoneum. They have serous membrane. What is serous membrane? It is simple squamous and areolar connective tissue. So serous membranes have simple squamous and areolar connective tissue. And these peritoneum, they have two layers. The inner one is called the visceral peritoneum, and the upper one or the outer one is the parietal peritoneum. So basically, this is, say, an organ. Okay, this is an organ. Say ileum. Okay, ileum of the small intestine. Now I'm going to show you. Take a different color. Yeah. So right on top of the ileum, you can see a layer of protection or covering. That is called visceral peritoneum. Visceral means internal. This membrane remains just sitting or affixed on top of the organ, sitting right on top of the organ. That's a visceral membrane, visceral peritoneum. Then there is a cavity like that, and this is the parietal peritoneum, the outer covering. We'll give it a different color. This is the outer covering, parietal peritoneum, and this is the one that is attached to the abdominal wall. So this kind of an arrangement is called as a peritoneum, and this kind of membrane that includes a visceral and a parietal peritoneum. So this plus this equals to a serous peritoneum. Okay, so the organ would have right on top of it a layer that is called visceral, and then after the cavity, this is the cavity. After the cavity, there would be another layer that is the parietal peritoneum, and the parietal peritoneum would be the one that is attached to the abdominal wall. Example of such an organ is the ileum of the small intestine, and this is called intraperitoneal organ. That means it's within the peritoneal cavity. Let's look at a retroperitoneal one. If it's a retroperitoneal one, then it's the abdomen. This is the kidney. It's directly attached to the abdominal wall by adventitia, and that is fibrous connective tissue. This is not within a peritoneal cavity. This is an example of retroperitoneal. This is intraperitoneal, within a peritoneal cavity. This is retro, outside the peritoneal. Both are attached to the abdominal wall. This is by fibrous, this is by the serous membrane. Any questions? We have 156, 155 slides. So sometimes I will have to move a little fast. If I go fast, stop me and ask me to repeat. But do not leave the class without understanding the topic. And that's more important for me. You need to understand everything. If you do not understand, stop me and ask me, but please learn something. So here are examples of all the peritoneal cavities that you can see. So for our heart, we have the pericardial cavity. This is the pericardial cavity. For the lungs, we have the lung pleural cavity. Sometimes a person if a person has lung cancer, or not only lung cancer, some kind of ascites or some kind of water leakage, it would cause inflammation, especially during the COVID pandemic. A lot of people had fluid accumulated in their pleural cavity, pleuritis and all pleurisy. These are situations where the pleural cavity is infected. Then we have, these are the retroperitoneal, which are outside. They are outside the peritoneal cavity. And here it shows you the visceral peritoneum right on top, sitting right on top of the organ over here. This is the peritoneal cavity, the whole one, and showing you the organs that are inside. Now look, retroperitoneal, this one, this guy is outside the blue line. The blue line is the peritoneal cavity. So intra versus retro. Intra, organs that can move. Retro, they should not move. These are within the peritoneal cavity. These are outside the peritoneal cavity. When an organ is enclosed by serosa, I already showed you what is a serosa. A serosa would have a visceral layer and a parietal layer. So visceral is right on top of the organ. This is the organ. This is the organ. So this is the visceral layer. A little on top of that is the parietal layer. So that is a serosa layer. And in case of the retro, it is when an organ lies against the dorsal body wall and it's covered by peritoneum on the ventral side only. So to the abdominal wall, you won't find any attachment. That means this is the abdominal wall, dorsal side. This is the ventral side. If the organ is here, the retroperitoneal organ, then it's attached to the dorsal wall of the abdomen by the adventitia, and just on the ventral side there could be a peritoneal cavity with other organs. That's what it means over here. Now, who are the examples? Stomach, liver, parts of the small intestine and large intestine. This is important. So what happens is people usually write, oh, small intestine, that is intraperitoneal. No, parts of it. So who are the parts? Some part of duodenum, jejunum, ileum, some parts of the large intestine like cecum, appendix, transverse colon, sigmoid colon. These are intraperitoneal. What about the ascending colon? What about the descending colon? They are not intraperitoneal. Okay, some part of the duodenum. The remaining part is not intraperitoneal. So retroperitoneal, duodenum, some parts of the duodenum, ascending colon, descending colon, head, neck, body of the pancreas. Remember, the tail of the pancreas is intraperitoneal. The head, neck, and body of the pancreas are retroperitoneal. For duodenum, the initial part, it says some part, I'll just tell you the initial part, the first part is intra. The remaining part of the duodenum is retroperitoneum. Some parts, that means the remaining part are retroperitoneal. Parts of the large intestine, that means ascending and descending, not the rest. Transverse and sigmoid, they are all intra. Rectum, kidney, ureterus, suprarenal gland or the adrenal gland, these are all retro. So this page is very, very important. You would be tested by each name: sigmoid colon, intra or retro; ascending colon, intra or retro; kidneys, intra or retro; initial part of duodenum, intra or retro; tail of the pancreas, intra or retro. So make sure that you understand what is intra, what is retro, who are inside, who are outside. Any questions? So this is a mnemonic that you can remember: pucker, or you say sad pucker, pancreas except tail, ureterus, colon, ascending, descending, kidneys, esophagus, rectum. All of these are retro. So we go to the next one, that is mesenteries, folds of tissue that extend from parietal peritoneum to the visceral peritoneum. So basically, mesenteries are tissue connections between the, I showed you just now, one is parietal peritoneum and what is visceral peritoneum. I showed you that here is an organ, right on top of it, the tissue that covers it is the visceral. And then a little far from it, that sits on it, is the parietal. This connection, the folds of tissue that extend from this to this, that would be the mesenteries. So who are the mesenteries over here? Falciform ligament that connects the liver with the diaphragm. Anterior peritoneal wall to the liver, also located between the right and left lobes of the liver. Lesser omentum, that's the ventral mesentery, extends from lesser curvature of the stomach to liver. So here is the liver. Okay, and here is the stomach. Now the stomach has a greater curvature and a lesser curvature. This is convex side, this is the greater curvature, and this concave side, that's the lesser curvature. All of these arteries and veins and all of these, they enter mostly through the lesser curvature part. Now you would find that this connection, this is the liver, this is the liver with the bile and everything. This is the liver. So the connection between the lesser curvature of the stomach and the liver, the spread out over here, this connection would be the lesser omentum, which is spreading between this one and this one. Here is the liver, here is the lesser curvature of the stomach. So we can call this connection, this flap or mesentery fold of tissue, this fold of tissue, we can call it as lesser omentum. The greater omentum, mesentery that extends from the greater curvature of the stomach and loosely covers the small intestine like an apron. So if you do dissection of a cat, when you open the cat's abdomen, you would find something like this, a big apron or a big cloth or a big umbrella spreading. That is the greater omentum. That's the fold of tissue or the mesentery that extends from the greater curvature of the stomach towards the small intestine. This is the intestine. So between this part and the intestine, this one spreads like a big umbrella, and that's the greater omentum. So this diagram would show exactly the locations of the lesser omentum, the mesentery proper. Here it shows the greater omentum. It's just labeled over here, not so diagrammatically clear or apparent. See the connection between the liver and the stomach's lesser omentum, lesser curvature. This is the lesser omentum. So here are some review questions for you. Peritoneum, that is the serous membrane. It is either intraperitoneal or retroperitoneal. So by now you know what is the peritoneum, what is the serous membrane, what is intraperitoneal, what is retroperitoneal, what are mesenteries or folds of tissue, where is the lesser omentum, where is the greater omentum, what is the falciform ligament between the, you can find the liver, between the liver parts that you would find the falciform ligament, mesocolon, mesentery proper. Mesocolon means the membrane that is connecting the colon. We'll go to the large intestine, then I'll show you exactly where the colon is. So the digestive system anatomy, macroscopic and microscopic parts. So we'll go to the microscopic parts now, or the histological layers, the tissue layers. We begin with the, first of all, the lumen right here. This is the lumen. We begin with the lumen. That means the food or the chem or the food mixed with the chemicals is called as chem. CHYMA. We call that as chem. So the food with all the enzymes and gastric enzymes mixed up, churned, it comes out, and this is the lumen, the canal or the tube through which it comes. So the food directly would be in contact, okay, with the layers. And here are the different layers. It's like one layer on top of another, like this. The histological structures are arranged like concentric rings, okay. So we'll begin with that. So first of all, I'll show you where is the lumen. Right here, this is the lumen. First one is mucosa. After that is submucosa. Then comes the muscularis externa. From the word muscularis, you can know that it has something to do with muscles. And then serosa layer. We can also see a bunch of nerve, artery, and veins. And the green color indicates the lymphatic vessel. So red for the artery, blue for the vein, yellow for the nerve, and the lymphatic vessel. All of them entering the nerves, innervating and the glands, these are the glands that are present. So we are going to read about individually about each one of them in detail. I'm just showing you the diagram. Intrinsic nerve plexuses are there. So these yellow lines that you see, these are nerve plexuses that send the impulses to the muscles to undergo peristalsis movement or wave-like contraction, so that the food can be mixed with the saliva and the food can move, be propelled in the forward direction. So we have considerable muscular movement and contraction, and that will not happen unless there is, nerve impulse. So myenteric nerve plexus is there, submucosal nerve plexuses. These work to bring about the contraction of the muscles. There are glands over here in the submucosa, and then mucosa layer, is consisting of the epithelium, the lamina propria, or the connective tissue. Muscular is mucosa. Then is the submucosa. Then comes the muscular is externa. So in the submucosa layer, you would find a lot of glands. Then comes the muscular is externa part. The muscular is externa consists of the longitudinal muscles and the circular muscles. Right here, it's not so clear over here, but these are the muscles, longitudinal and circular muscles. And then the last one is the serosa, consisting of the epithelium, simple columnar epithelium, and then the connective tissue. Okay, so you have to remember what are the histological layers. So starting with the lumen, you would have mucosa, submucosa, muscularis externa, and serosa. So looking at that diagram, now let's go in detail. The mucosa, which is closest to the lumen. So if this is the wall, this is the lumen. The food is going to come through this, so lumen, and right sitting on top of it, right here, that would be the mucosa layer. M for mucosa. Okay, and it's consisting of epithelium, that is simple squamous, simple columnar epithelium, then lamina propria, that is connective tissue, areola, and muscularis mucosa, or smooth muscle. Why is it smooth muscle? Why is it not skeletal muscle? Because when the food travels through the digestive tract, it is involuntary. It's not under our conscious control. If it was conscious control, then we would have decided when we want the digestion to happen and when we want to stop the digestion. Well, it's not under our control. So it's not skeletal muscles. It's smooth muscle, and it is involuntary. It's not under our conscious control. And that is why it is smooth muscle. So the mucosa layer consists of these three parts. The next one is submucosa. Use a different color. So the next one is submucosa, this one. So it has dense irregular connective tissue, and the submucosa is the one which is housing or hosting or keeping the nerves. So you have the enteric nervous system, Meissner plexus. This is important for your quiz question, and it controls the movement of the mucosa and glandular secretions. So muscle contraction, smooth muscle movement, glands will secrete. How will the glands secrete? Unless there is a motor nerve telling the gland to secrete. So there must be an efferent or a motor nerve that tells the gland to secrete. So that information comes through this plexuses, through the enteric nervous system. So submucosa is important in the sense that it will have the nervous system or enteric nervous system to instigate or to order the muscles to contract or the glands to secrete. So next layer sitting on top of that would be the muscularis externa, and it is important because of peristalsis. It consists of inner circular and outer longitudinal muscles, and it does peristalsis. That means contraction of the inner circular and shortening of the outer longitudinal muscles. So you have this wavy contraction motion that is called as peristalsis. Like you take a toothpaste and you press it and the paste comes out like that. The food passes through the alimentary canal thanks to this wall contraction and relaxation. Between the above two is the Orbac plexus that is also there. There's another plexus, and it controls the peristalsis, carries the impulses for smooth muscles of muscularis externa. So muscularis externa will contract, the smooth muscles will, the circular and longitudinal smooth muscles will contract when they are provided nerve impulses using the Orbac plexus. So that is, where is it located? Between the above two, just between these two, just above that would be the Orbac plexus. Then lastly, the last on top of them is the serosa or the adventitia. So if it is a peritoneal, then it is serosa. If it is a retro, then it is adventitia. So that's why it is written this or this. So serosa, serous membrane, that is areolar tissue topped with simple squamous epithelium, and it covers the muscle layer along most areas of the GI enclosed within the peritoneal cavity. So this guy is within the peritoneal cavity. If it is retroperitoneal, then it cannot be serosa. Then we will call it as adventitia, and that would be a fibrous connective tissue consisting of dense collagen fibers, and they will simply take this whole thing, stomach, and attach it to the wall of the abdomen with the help of fibrous adventitia. The serosa versus adventitia. So serosa is visceral peritoneum, as you know, and it is areolar connective tissue with simple. Are you guys sending any text messages? Okay, because this screen is staying above, so I can't see any messages. So serosa is areolar connective tissue with simple squamous cells on the surface. It secretes a watery fluid, and it I asked a question. Okay. Oh, I'll show you. 1 second. Let's go back there. Submucosa, this layer, it has these enteric nervous system, because it's going to control the glandular secretions. Okay, it will help in the, I'll show you over here, right here, submucosa. It's sitting right here, submucosa, this guy. Can you see this one? This is submucosa right here, this layer that has a lot of glands, and it has the enteric system, the nerves that will help in the glandular secretion. So coming back to the adventitia, organs that are outside of the peritoneum cavity, and they will have the adventitia, they will not have the serosa. That means the organ, this is the abdominal wall directly attached by fibrous connective tissue, and that is called as adventitia, made up of fibrous connective tissue. Control of the GI organs. So who controls these organs or these muscle layers? It always go back to the nerves. Okay, so this part is important. The endocrine system, with the help of hormones, that will help to control the digestive system, because we have gastrin hormone. Not only gastrin hormone, there are a series of other hormones, 7 or 8 different types of hormones that will play a role in the digestion process. But apart from the hormones, the next important part is nerve. So that's why we always talk about neuroendocrine. Why? Because nerves and hormones both will help in the digestion process. So hormones, we'll go to that in detail when we go through the biochemical aspect. Now let's look at the nervous system. The autonomic nervous system, okay, before that, the nervous system involves intrinsic or enteric nerves and extrinsic or sympathetic and parasympathetic. So what is the difference between intrinsic nerve and extrinsic nerve? From the word intrinsic and extrinsic, you can understand that internal and external. That means some nerves are embedded in the GI tract, like the plexuses we talked about. Uh, these nerves or plexuses which are embedded, or it's like inbuilt. You have a inbuilt speaker system in your mobile phone, inbuilt speaker. But then you also want to add a Bluetooth or an external device. That's an external. Both may do the same functioning, or external microphone, same functioning, but one is intrinsic, embedded inside the mobile phone, and one is external. So same way, we have an enteric nervous system which is embedded in the wall of the stomach or the GI tract. I just showed you the Urbach plexuses. These are the inbuilt enteric nervous system. The extrinsic nerves, they connect to the brain and the spinal cord and to the medulla and all those places, and they are extrinsic or external or outside the GI tract, and they could be sympathetic or parasympathetic. Parasympathetic is rest and digest. As you know, sympathetic is the one, fight or flight. It's very, very important that we have a balance of the parasympathetic and the sympathetic nervous system. I mean, when I say balance, it means that they should be working at optimum. If you have um, an imbalance of these nerve system, you know what is called as anxiety poop. Have you ever gone through that? Anxiety poop is like, you here in the morning, your exam results are coming out, okay, you just have to rush to the restroom. That's anxiety poop. Or you can say somebody heard something, a bad news or something. They get really scared, they have anxiety, and they want to go to the restroom right away. So that's how our digestive system is connected to the nerves. Okay, so parasympathetic indicates rest and digest. It, remember, increases the activity of the GI tract. Please note it. It increases. Question for you in the exam would be, does the parasympathetic system increase or decrease the digestion or the activity of the GI tract? Or which system, is it sympathetic or parasympathetic, increases the activity of the GI tract? Mostly it works through the vagus nerve. Vagus is the 10th nerve, the most important, the mixed nerve, and it's very, very crucial, very, very important. The next one is sympathetic, fight or flight. It decreases the activity of the GI tract. Okay, why does it decrease? Because if your sympathetic system is activated, like fight or flight mechanism, your energy is focused in that direction. Your energy is focused in that fighting or flighting mode. So that takes the prominence, and your food digestion would be not the focus of the digestive system of the brain at that time. It would rather pay attention to the fight and flight mechanism. So that means it's going, if that system is activated, like you are in a fight mode or you are in a flight mode, you're very scared or you're very flight mode, your digestion would be slowed down. Okay, your digestion is going to be slowed down. So without proper digestion, the whole focus and the energy would be towards fighting or towards flight, running away, not the digestion. But when you are sleeping and you are relaxed, then your rest and digestion system is activated. It's parasympathetic, and it's going to go through the normal digestion process. The mind-trick system, that is influenced both by sympathetic and parasympathetic. This is the intrinsic one. These are the extrinsic. This is the intrinsic or the inbuilt one. And this is influenced by sympathetic and parasympathetic. Both control it. Intrinsic controls the motility and secretion. It includes the myenteric and the submucosal. Your friend just asked about the submucosal plexus. This is located in the mucosal layer, and they control the chemo and the stretch receptors. These chemo and stretch receptors are controlled by the enteric system. Enteric is influenced both by para and sympathetic system. Next, we will go to the mouth. Any questions? Okay. So mouth, it consists of the anatomical structures, teeth, and salivary glands that provide saliva, that has the salivary enzymes that would help in chemical digestion. As I already told you, mechanical digestion is mastication or chewing, breaking the food into small bite pieces. But chemical digestion is different. That involves saliva or the enzymes that break down the complex food into simple ones. So if you look at the mouth and all its parts, labia, the lips are called as labia. Teeth, we have the mouth cavity or the oral cavity. Tongue, the uvula. Okay, the uvula is right. If you, it doesn't show over here, but the uvula is right here, like this. It kind of hangs like this kind of structure. That's the uvula. It helps in our swallowing. It helps in speech. Okay, and to some extent, people who have sleep apnea, it also involves the uvula. One pharyngeal tonsil or adenoid tonsil contains the immune cells. As you know, they fight against bacterial infection, the tonsils. And then pharynx, components which are part of the GI tract are the oropharynx and the laryngopharynx. So pharynx, so let me show it to you. Okay, so this is the mouth. The pharynx would be the nasopharynx, the oropharynx, and the laryngopharynx. Out of these, this particular oropharynx would be the part, and the laryngopharynx would be the part of the digestive system. The nasopharynx would be part of the respiratory system. Then lingual frenulum, if you lift your tongue right here, under the tongue, you will find a structure, right? This is your tongue. There is a structure like a membrane. Okay, this is the tongue. And here's a membrane, just as if it is, when we lift our tongue, we can see that perpendicular membrane. That's the frenulum. It stabilizes the tongue. And then hypoglossal 12, this is the nerve, that hypoglossal, that innervates the tongue. So question for you over here, which nerve provides impulses to the tongue? That is hypoglossal 12, the 12th nerve. Most common infection, Streptococcus pyogenes, that is a group A Streptococcus, and mostly you'll find it affecting these tonsils. And sometimes doctors say if antibiotics don't work, go for surgical excision. So looking at the parts of the mouth, this is the soft palate. It helps in holding the food. Then we have the palatoglossal arc. The roof of the mouth is called as the palate. This is the hard palate. This is the hard palate. And then the soft palate. Here's the uvula. The palatine tonsil right here. This is the palatine tonsil. The tongue looks like a big fleshy organ over here. Since this is the mouth, you can see here is the oropharynx. Okay, so the back of the mouth, this channel, the entire channel, the part over here, the back of the mouth, that would be exactly the oropharynx right here. Then we have the lingual tonsil. Lingual means tongue, and right at the base of this tongue is the lingual tonsil. The hyoid bone, laryngopharynx. This is the laryngopharynx. Okay, and right here, the extension of the laryngopharynx would be the esophagus. So on the ventral side or the anterior side, you have the trachea. So that means the respiratory chamber, the respiratory passage. This is the respiratory passage, ventral or anterior to the digestive GI tract. Okay, so assuming that this is the nose, so if this is the oropharynx, we have the nasopharynx somewhere here. This is the nasopharynx, oropharynx, laryngopharynx, and then down there is the esophagus. We are not going to talk about the nasopharynx because it's part of the respiratory system. Now, looking at the anterior view, this is the lower lip, and here's the uvula, the soft palate, hard palate. Palate will help you to hold the food and helping in chewing. Gingiva, or the gums, they help in they protect structurally, and they have also some function to fight against bacterial infection. So gums, healthy gums will look pink in color. If it is infected, it will look red. Then this one right here is the interlabial frenulum. This is the lingual frenulum that I was talking about, that kind of holds the tongue in shape. Okay, this spot right under the tongue, that's lingual frenulum. Okay, we'll go to the next slide. Mouth and pharynx. So mouth, it is stratified squamous epithelium. Whereas the digestive system, when we talked about the stomach wall, it was simple columnar. But over here it is stratified squamous. Why? Why do you think it is stratified squamous? Stratified means it is multi-layered, not single layer. Well, we eat food, and our food has sharp things, sometimes crispy things. It has chemicals. We eat all kinds of stuff, and so our mouth provides protection against abrasion. So protection against abrasion, and that is why we have stratified squamous epithelium. So that's your answer for why. Generally, it is non-keratinized. However, certain areas are keratinized. Like, for example, palate, it is keratinized, extra layer of protein. And why? Because it prevents desiccation. Because our mouth, when we pant, is exposed to the atmosphere, so we can lose water vapor, we can lose moisture, we can lose water. So to prevent dehydration, it is kind of keratinized with extra layer of protein. It provides dryness, and so water will not evaporate out of our mouth. So that's a natural mechanism of protection. We have connective tissue in the form of lamina propria, and that's areolar connective tissue. Uvula, as I said, helps in swallowing the food, also in speech. Gingiva, or the gums, they are made out of mucosal tissue, and they are barrier against infection. They're also structural support because they help the teeth to lodge into the gums successfully. If the teeth looks like it is dislodged from the gums, that means there is bacterial infection, and that's why the gums are receding from the teeth. Hard palate, maxilla and palatine bones. Soft palate helps in speech and swallowing and breathing. What are the salivary glands? These are the accessory digestive structures, those that help, the helpers. They are exocrine. Why they are called exocrine? Because they have a duct. If they have a pipe for secretion, like this is a gland, it has a pipe, and then there is a secretion. This is called as a duct or a pipe, and such glands are called as exocrine. If it is endocrine, this will always secrete an enzyme. Exocrine will always secrete an enzyme. Whereas if it is an endocrine, then it's going to secrete hormones, and it is ductless, no pipe. So now if you look at parotid gland, long duct opens into the mouth. So here is the parotid gland, okay? And look at the duct, the parotid duct, quite a big pipe, quite a big channel, and it has a watery serous solution of saliva, no mucus production, and its infection is called as mumps, where this one kind of inflammation and swelling happens. The next one is submandibular, and it is a combination of serous and mucus secretion. Okay, so serous is the watery, like water kind of thin secretion. Mucus is more translucent kind of secretion. So that is submandibular. So let me clear up this area for you. Okay, so we can see the mandible means the jaw area. So submandibular means below the jaw. Then we have the sublingual. Lingual means tongue. So below the tongue would be sublingual. Okay, and below the jaw would be the submandibular duct. We have the masseter muscle over here, important because it helps in mastication or chewing. This is the mandible or the jaw, and this is the parotid, and here is the duct that supplies the secretion of saliva. So parotid would be serous, keep in mind. Submandibular would be serous plus mucus. Sublingual is mucus. So this is S, this is M, and this is S plus M. So how does the salivary gland look? It's an exocrine. As I told you, they have a pipe or a duct. They look like grape-like cluster of cells, and or acinus or alveolus, like a pocket of like grape-like structures like this, a bunch of grapes like this. And serous is watery secretion, appears dark. Mucus acinus appears white, and the ducts carry product to the mouth mostly through this epithelium, simple cuboidal epithelium. So the question would be, the salivary glands have what kind of epithelium? That is simple cuboidal epithelium, because in cuboidal particular epithelium, they are going to secrete or push the salival secretion with the enzyme through the duct into the lumen. Saliva, what is its composition? It contains the enzyme salivary amylase. This is important. And carbohydrate undergoes hydrolysis with the help of amylase enzyme to break down the carbohydrate. And it also contains, saliva also contains 98% of water, mucus, antibacterial lysozyme to kill the bacteria, and then antibodies A. So and the iron, sodium, potassium, bicarbonate. The question for you over here is, what name the antibody that is in saliva? That is IgA. Don't write anything else. If you just write antibody, you will not get any marks. So you have to write specifically IgA, immunoglobulin, that is present in saliva. And what is the function of lysosome? Antibacterial, kills the bacteria. What is the enzyme present? Salivary amylase. On what does it act? It acts on carbohydrate. By what process carbohydrate digestion happens? Carbohydrates will be acted by salivary amylase, and with hydrolysis, they will make use of water plus water, hydrolysis, to break it down into glucose. Control of salivation. So facial and glossopharyngeal nerves, they will control or regulate the salivation process. That means these nerves will cause the glands to secrete. That means these are motor nerves, efferent motor nerves, not sensory, not afferent. They are bringing the information from the brain to the glands, and they are causing the secretion of saliva. So food in the mouth, or the smell of food, also acts as a good sensory mechanism to activate those, and through the parasympathetic nervous system, it will increase the salivation. So question for you here, which mechanism, which system of nerves causes or increases salivation? Is it parasympathetic or is it sympathetic? It is parasympathetic. And we have our control center of the salivation also in the medulla oblongata. Next, we'll go to the tooth. Any questions? Okay, so looking at the tooth, the first one is enamel. It is the hardest substance in our body. So name the most toughest or the hardest substance in our body. That is enamel, and it's made out of calcium phosphate. The next one is dentin. It's a calcified tissue, and this is acellular, this is cellular structure. Then we have the pulp cavity right here. This is the pulp cavity, and it has the blood vessels, nerves, and connective tissue. So when you have cavities or any bacterial infection in your tooth, initially there may not be a tooth pain. You just see a black spot. But if the bacterial infection, eats up that area and destroys that part, but the day it touches the pulp cavity and the nerves, The day it touches the nerves, that day you're going to have a toothache. So if you have a tooth, this part is already eroded, this part is already eroded, and now it's just touching this part. And if you don't take care of your tooth, then very soon it's going to even go and affect the root area and completely dislodge the tooth. So periodontal ligament, these are collagen fibers, and they attach the tooth to the alveolar socket. So here's the alveolar socket, and the periodontal ligament does a good job here, right here. This is the periodontal ligament. It will fix or attach the tooth to sit on the alveolar socket. This is the jawbone. This one would be the alveolar socket, and the tooth comes out of it, and the connection between them is the periodontal ligament. Gingiva, that is stratified squamous epithelium, and it also contains connective tissue lamina propria. Why is it stratified? I have already told you, because chewing and mastication and all of these food chemicals will enter. So we double layer, triple layer, multi-layer protection. That's why it's stratified. Gingiva is the gum. Then we have the crown. This part is the crown that is visible to our eyes. Then the root or the base, this is the middle part, is the neck. Deciduous teeth is the baby teeth, and they consist of 4 incisors, 2 canines, and 4 molars. The permanent teeth has the premolars as well. Next, we come to the pharynx. The pharynx consists of, like I told you, the nasopharynx, the oropharynx, and the laryngopharynx. So 3 parts: the nasopharynx, and the oropharynx that's connected to the mouth, and the last third part is the laryngopharynx. For the oropharynx, it is both air and food, and the nasal, as you know, is part of the respiratory system. We'll see it later on. Now, the histology, if you look at the nasopharynx, is made out of pseudostratified ciliated columnar. That's for the respiratory system. The oropharynx and the laryngo both are stratified squamous. Why is it stratified again? Because the food will pass through it through the difficult process of swallowing. Okay, so that's why stratified squamous, multi-layered, to provide toughness against abrasions. And then muscle, it has longitudinal muscles, and it also has circular muscles. Now next is the esophagus. So we done with the mouth and then the pharynx, and now let's go to the esophagus. The esophagus, it consists of the upper esophageal sphincter, UES, upper esophageal sphincter. A sphincter is like a valve. I will show you how it works. Then we'll talk about the esophageal hiatus, the lower esophageal sphincter, and we'll talk about a little bit about their wall, their layers. We'll talk about peristalsis, the wave-like motion that helps in pushing the food down. What kind of digestion happens in the esophagus? Is there any chemical digestion? No. Is there any physical digestion? No. Basically, it helps in pushing the food down. And what is the muscle that is present in the esophagus? That is cricopharyngeal skeletal muscle. Why is it skeletal muscle? Why cannot it be a smooth muscle? Because the esophagus is involved in deglutition. Deglutition means swallowing. Swallowing is the only main job of esophagus. Deglutition. Be watchful of the spelling. So now, if swallowing is a process where we have to push the food down, so some part of it is voluntary, and towards the end you would see when it is connected to the stomach, it becomes gradually involuntary. But the part of the esophagus that is voluntary is done by cricopharyngeal skeletal muscle, and it has vagus nerve innervation. So when we study the digestive system, you will find that each part that is involved, mouth or pharynx or esophagus or stomach or small intestine, all of these involve muscles and nerves. As you go from the upper part, it is skeletal muscle, which we can control. As you go deeper down inside the GI tract, it converts into smooth muscle where the digestion happens and we are not aware of it. We cannot control the digestion. If I take a piece of cake in the morning, I cannot decide that I will digest it at 2 p.m. No, I cannot. My body will automatically digest it without my knowledge, and that is involuntary. And when it happens without my knowledge, involuntarily, it is smooth muscle. What I can control is the swallowing or the deglutition. And that is why I have skeletal muscle that I can control. I will talk about the esophageal hiatus. So, esophagus, it is located posterior to the trachea, so backside of the respiratory tract, but anterior to the vertebral column. So you can see over here, this is the trachea, this is the esophagus, and back side would be the vertebral column, front side is the respiratory. So in the middle part is the esophagus. It's 12 inches long. It consists of upper esophageal sphincter, a lower esophageal sphincter. These are like valves, and then esophageal hiatus, a whole diaphragm in order to reach the stomach. So right here, this is the esophagus, and as you can see, as it goes down, this is the diaphragm. Let me use a different color. This is the diaphragm, okay? The membrane that separates the thoracic from the abdominal. And so it kind of makes a puncture over here, it makes a hole over here, and then esophagus goes down and connects to the stomach. So this part where it punctures the diaphragm and goes inside, that's the hole in the diaphragm. That's called esophageal hiatus. Sometimes the hole tends to swell up, inflamed, and becomes bigger, and then the stomach tries to come up through that hole like a hernia, and people suffer from it. Now, esophagus, it has an upper esophageal sphincter, a valve, and a lower esophageal sphincter. Basically, these are gate or valves, as you can call it. So when the sphincter will relax, when the sphincter will open up and relax, food will go down. Once the food has gone down, the sphincter is going to contract and become tightened and smaller. So food cannot return back. So there would not be any reflux, because once the food goes down, the sphincter which had become relaxed will now become contracted. It will close the sphincter. On the other hand, the lower sphincter, which was contracted, it's now going to expand and open so that the food can go into the stomach. Once the food goes to the stomach, this sphincter is going to contract and close. There's basically a gate. The sphincter is like a gate. It will open, allow the food, and then it will close. So the food cannot return back. We have two gates, one in the upper part and one in the lower part. Control of esophagus, peristalsis regulated again by, remember the nerves, the enteric nervous system, Auerbach's plexus or the myenteric plexus, between the inner circular and outer longitudinal muscles. They are both influenced, as I told you, by parasympathetic and sympathetic, and they respond to stretching. If you keep on eating a lot of food, okay, so it kind of stretches the wall of the esophagus, and you can see it, how the peristalsis moves the food. Sometimes you can feel it. You can feel the food moving through the esophagus when you swallow. And then response to stretching enhanced by vagus nerve. There is no significant chemical or mechanical digestion that occurs in the esophagus. So esophagus is basically a channel for the food to pass through it. Clinical significance. Two things that may go wrong. Though there is no digestion in esophagus, two things that can go wrong. One is a reflux, and it is mostly in small babies. They drink milk and they have a reflux where they vomit or puke out curd, okay, because their digestive system is not well developed yet, or their sphincters are not closing well enough yet. So they would have a milk reflux. So caused by, and then you see babies, they just wake up crying because of the reflux, and caused by weakness or the lack of contraction of the lower esophageal sphincter. Acid from the stomach can get into the esophagus or up to the oral cavity. It can damage the mucosal lining. It causes reflux and heartburn. So if this two sphincters, the upper and the lower, if they don't function well, then from the stomach, chemicals can return back in the backward direction and even come up to the mouth or the oral cavity. And that gives us a bitter taste in the mouth. So that is one, reflux, GERD. The next one is the hernia or the hiatal hernia. So we know that through this hole, the esophagus enters, punctures and enters into the stomach. Now, sometimes there is an abnormal hole and it will cause acid reflux, heartburn, difficulty breathing. And these, if it is swollen and there is inflammation, it's called as a hiatal hernia. See the way how it is swollen, and part of the stomach begins to move up into the esophagus through that. And that is called as hernia. Three phases of deglutition or swallowing. One is through the mouth, that is voluntary. You can control the buccal or the chewing that you do in your mouth. The next one is also swallowing through the pharynx, that is voluntary. Then esophagus is important because the first upper part is voluntary. The middle part is mixed, partially voluntary and partially involuntary. And the lower esophagus is absolutely involuntary. So mouth, pharynx, esophagus, stomach. So this is voluntary swallowing. From the esophagus, you have the first part as voluntary, the second part is voluntary and involuntary, and the third part is completely involuntary, and then small intestine is involuntary, and the large intestine is involuntary. And then at the anus, again, there are two parts. The first part is involuntary, and the last part of defecation is voluntary. You decide when you want to go to the restroom. So, three phases of deglutition. Buccal, it is voluntary, make bolus of food and push it into the oropharynx. The soft palate will lift up and seal off the nasopharynx so that food will not enter into your nose. Okay, that's the mechanism. And the upper esophageal sphincter will contract. During the buccal phase, the tongue will press, the tongue will lift up against the hard palate, and it will cause the food bolus, which is mixed with saliva, to go into the oropharynx. Yeah. Now, in the pharyngeal region, what happens? The uvula and the larynx will rise to prevent food from entering into the respiratory passage. The tongue will block off the mouth, and the upper sphincter relaxes. It has to relax because it will allow the food to enter from the pharynx and to move towards the esophagus. So pharyngeal swallowing is voluntary, and the bolus passes through and stimulates tactile receptors, so swallowing reflex happens. Which nerves are involved? This is important. Trigeminal, 5th, and the glossopharyngeal, cranial, 9th. These are important, and the swallowing center is in the medulla oblongata. The epiglottis will fold. This is a protective mechanism. It will fold and close off the larynx so that the food will not enter into your air canal or your air passage, where your respiratory tract is closed, shut off. So food will be directed towards the esophagus. Pharyngeal phase, the tongue blocks of the mouth, the upper respiratory relaxes, allowing the food. So this is done. Last one is the esophageal passage. The contraction of the pharyngeal muscles forces the food down into the esophagus. Peristalsis will propel. By now you know what peristalsis is, a wave-like contraction, and it will propel the food into the stomach. So we go step 3, down from the esophagus down into the stomach. The upper esophageal sphincter will close after the entry. So once from the esophagus, once the food enters into the stomach, once the food goes down, this one, the upper esophageal sphincter, this is going to shut off. Once the food goes down, the lower is also going to shut down. Otherwise, reflux will happen. So we don't want the reflux, so both the sphincters will close. So here it shows relaxed muscles. Circular muscles are contracting. Peristalsis, by peristalsis mechanism, the food is going down. Okay, and these are the longitudinal muscles which are contracting. So alternate mechanism of circular and longitudinal muscles contracting. The food is going unidirectional in one direction, and right here this sphincter is right now closed. Why is it closed? Because the food is at this level. Once the food comes here, then the sphincter will open and the food will pass into the stomach. Once the food enters into the stomach, this sphincter will also close. Since the food has already come down, this is now relaxed muscle. Okay, so now this sphincter opens, the lower esophageal sphincter has opened, and the food is traveling into the stomach. Whatever I just explained to you, the passage of food or the deglutition, step 1, 2, 3, 4, and 5, how this valves open and close, and how this valve open and close, and how the food passes along is depicted step by step in this diagram. So now peristalsis versus segmentation. So peristalsis would be, you can see the arrow unidirectional, alternate contraction of the longitudinal and circular smooth muscle, and what you get is the chyme, food that is mixed with saliva and all the gastric juices is called as chyme. So now chyme, we'll not call it food anymore, we'll call it as chyme. Chyme is moving down through peristalsis, alternate contraction of circular and longitudinal muscles. So see, relaxing, contracting, how it is moving in a propulsive forward unidirectional manner. Now, in case of segmentation, it is so contracting circular muscles and relaxed longitudinal muscles, but they are in a back and forth churning motion. This is more like a churning motion, circular churning motion. This is unidirectional. This is back and forth, like this kind of motion. Why this is important? Because why is important? Because this back and forth churning motion will help in mixing the enzyme with the chyme. So these are the sphincters that will be used. Pharyngeoesophageal sphincter, UES. If this is the esophagus, here is the pharynx, esophagus, stomach. Okay, so here is the UES. Okay, and the LES, lower esophageal sphincter, and the upper esophageal sphincter. And why is it called cardiac? Because the stomach's first part is the cardia part of the stomach. The stomach has different regions which we'll study: cardia, fundus, body, pylorus. So the first part of the stomach is called cardia. So the sphincter is right sitting on top of the cardia. That's why it's called cardiac. Esophagogastric sphincter is the junction between esophagus and stomach right here. That's the lower esophageal sphincter. This one is upper esophageal sphincter or gate or valve between the pharynx and the esophagus. This is between the esophagus and the stomach. So lower and upper esophageal sphincter. So over here it shows you the exact location of upper esophageal sphincter between the pharynx and the esophagus. And here it shows you the position of the lower esophageal sphincter between the esophagus and the stomach. And hiatal hernia, that is when the swelling happens right here because of inflammation. Mostly sometimes in case of obesity also it happens. Or people having too much of reflux issues. So lower esophageal sphincter is closed. It shows over here. This is the diaphragm. And in case of GERD or gastroesophageal reflux, right here, you see how the food particles are returning in the backward direction because the sphincter did not close. It was open and it allowed the reflux to happen. Now coming to the wall, it has again same mucosa, submucosa, and muscularis externa, circular layer, longitudinal layer of muscles, adventitia or fibrous connective tissue that is connected to the wall of the abdomen. That's the histology of the esophagus. Next, we go to the stomach. Any questions? Okay. If you do not have any questions, we will go to the next one, that is stomach. The stomach is also called as the gaster, and it is J-shaped in structure. And we are going to talk about the chemical digestion. We are also going to talk about the physical digestion and the control, that is that the nerves that control the stomach. So the stomach is divided into the cardia. As I told you, the first part, this is the fundus region, the roof of the stomach, the main body, and the last part is the pylorus. So cardia, fundus, the roof, the main body, and the last part, which is called as the pylorus. The pylorus is divided into the pyloric antrum and the pyloric canal. There is a sphincter or a valve at the end of the pylorus. Here starts the SI, or the small intestine. So you can see duodenum. Duodenum of the small intestine is right here. So the connection between the stomach and the small intestine, right there, you have this valve which is called as the pyloric sphincter. It regulates when it opens or relaxes, the food will go into the duodenum. When it is contracted, it cannot pass. Same way, between the esophagus and the stomach, you have the lower esophageal sphincter, LES. So question for you would be, mostly they ask a question that where is the lower esophageal sphincter? The lower esophageal sphincter is between the end of the esophagus and the beginning of the stomach. Where is the pyloric sphincter? The pyloric sphincter is at the end of the stomach and at the beginning of the duodenum or the small intestine. So please do not get confused between the position of the sphincters. Then the stomach also has folds of mucosa or submucosa. These are called as rugae. These rugae are, as you can see, these are folds of the wall. They are called rugae because they help in more expansion of the stomach wall. Maybe you liked some particular food and you ate a huge amount of the food. So this kind of foldings of the membrane, it will help in expansion of the stomach, or to accommodate the amount of food that we take in. So, and then here's the greater curvature of the stomach and the lesser curvature. The muscles that are present over here would be the longitudinal muscles. Then we have the circular muscles, and we also have oblique muscles. So three muscles. Muscularis externa has three muscles. Other parts have longitudinal, and circular. The stomach has longitudinal, circular, and oblique muscles. That explains how much it allows for expansion, for churning of the food in the stomach, for housing the food in the stomach. But that's why extra muscles. If we look at the wall, the mucosa would be simple columnar as usual, folded into gastric pits and gastric glands. So what's the difference between a gastric pit and a gastric gland? So pits would be like this. These are gastric pits, and at the end of the gastric pit you would have a gastric gland that would pour out the secretion into the pit. That's the difference between a gastric pit and a gastric gland. It also has lamina propria, connective tissue that is areolar in nature, and it has muscularis mucosa. You see, it is smooth, it is not skeletal, because you cannot control the digestion or contraction of your stomach. Can you voluntarily, willfully control your stomach digestion? No, you cannot. That's why it is smooth muscle. Then you have submucosa. It consists of artery, vein, lymphatic vessels, and loose connective tissue. We have three layers, as I told you, in the muscularis externa layer, that is oblique, circular, longitudinal. Remember, there are three muscles in the muscularis externa of the stomach wall, compared to others who have two muscles. Serosa, function of the mucus, it protects against acid and gastric enzymes. So the acid that is secreted in the stomach or the gastric enzymes, they themselves do not eat away the wall of the stomach itself. It doesn't harm the wall of the stomach. There are other reasons for it, but one of the contributing factors is because mucus is there. Mucus protects against the harmful action of the acid. What are the glands that are present in the stomach? So the cardia part, so if this is the stomach, this is the cardia, this is top is the fundus, body, and the last part is pylorus. So glands in the cardia would be mucus secretion. Glands in the fundus would be acid and enzyme secretion. Glands in the pylorus, this part, would be fundus and body, both. And then glands in the pylorus would be mucus, digestive hormones like gastrin, all of these would be secreted in the pylorus. Then we also have enteroendocrine cells. These are like sensors that release hormones, and they connect the brain and the gut axis. Like I told you about the anxiety causes you to go to the restroom, that is because of the enteroendocrine cells or the sensors that release hormones. So a lot of hormones and a lot of nerve stimulation or implication would, sorry, the screen has moved. A lot of nerve or hormone would control the whole process of digestion. So let's look at the cells that are present in the digestive system. Mucosa, specialized simple columnar cells. So first of all, goblet cells. This is very, very important, even from a lab perspective. In the histological slides, they will ask you to point at the goblet cell and to show you in the wall of the stomach lining. And it secretes mucus because mucus protects against acidic pH of the gastric juice. Gastric juice is secreted by the stomach for digestion, but the gastric juice that is secreted for digestion does not do any self-harm. It does not digest the wall of the stomach itself because of the protective layer of mucus. Gastric gland has these types of secretory cells, and they produce 1500 milliliter of gastric juice per day. That's the amount we produce each day. So parietal cells, they appear pink. Number one, parietal cells, they're very important because they produce hydrochloric acid. They secrete hydrochloric acid. It kills the bacteria, and most importantly, it activates pepsinogen. What is pepsinogen? Pepsinogen is an inactive enzyme, or also called as a proenzyme. Now that is activated because of HCl. Who secretes HCl? Parietal cells. Where are parietal cells present? In the wall of the stomach. They secrete HCl that converts pepsinogen into active pepsin. What is the work of active pepsin? This active pepsin will be helpful in the digestion. Later on, we are going to study about the chemical digestion. It will digest the food. So HCl produced by the parietal cells converts the inactive or the proenzyme or inactive pepsinogen into active pepsin. What if pepsinogen is not activated? If it is not activated, it will not work. It cannot work. It is inactive. It has to be activated, and that is done by HCl, and it works on by denaturing the protein. The second thing parietal cells secrete is an intrinsic factor, which is called as a glycoprotein, that helps in absorption of vitamin B12. Parietal cell also indirectly secretes HCl. How does it do it? Because carbon dioxide and water combine to form carbonic acid, and this carbonic acid that is formed in the parietal cells, it dissociates or it splits into bicarbonate and hydrogen ions, and the bicarbonate ions are exchanged for chloride ions from the interstitial fluid. So basically, let me show this to you by diagram. So basically what it does is, if this is a parietal cell, it has H2CO3, okay? And this one splits into bicarbonate ions and H+. And this bicarbonate ion goes out, and in return, chloride. Okay, chloride, it is exchanged for chloride from interstitial fluid. As a result, now when chloride comes in, hydrogen and chloride completely form HCl. or hydrochloric acid, and they provide the acidity. So you may ask me, how did bicarbonate first of all end up in parietal cells? That's because of a combination of carbon dioxide and water, of hydrogen. So that formed carbonic acid, and that split into bicarbonate and H plus, and in exchange for bicarbonate we got chloride, and then now H and chloride become friends and they become HCl, acidic medium, and this acid can kill bacteria, and it mostly works on pepsinogen, which is in the inactive enzyme state or a proenzyme. So now HCl works on it, converting it into active pepsin, and the active pepsin can now work on the protein and break it down into amino acids, and that's how digestion is done. So parietal cells, they provide an acidic medium, and that's how it's important. The chief cells, they are the next group of cells. They appear dark blue, and they are the ones who secrete pepsinogen or inactive proenzymes. Proenzymes that are inactive are called as zymogens. Okay, they are inactive. Somebody has to activate them. In this case, HCl secreted by parietal cell goes and activates them. Next is enteroendocrine G cells located in the gastric glands. So enteroendocrine G cells located in the gastric glands, and it is an endocrine cell, and they secrete hormones. What are the hormones that are secreted? Number one, gastrin is a hormone that regulates the activity of the stomach, and it stimulates the parietal cells to secrete more HCl. So gastrin, remember, causes more HCl production by acting on parietal cells. That means gastrin increases acidity. Gastrin increases acidity, causes contraction of the gastric wall, helps to mix. So point number one that you should remember is that gastric hormone increases HCl production. How? Because it works on the parietal cells and causes them to produce more HCl. I showed you the mechanism how more HCl is produced by the parietal cell. Next is enterochromaffin cells or EC cells, and they secrete serotonin. If you ever heard about dopamine and serotonin, so these are serotonin in this case secreted by enterochromaffin cells, and it's important in chemical and mechanical stimuli in the gastric lumen. It works on it gets activated, especially in the brain gut axis. We talked about anxiety poop, where people have to go to the restroom when they hear something bad news or something scary. So serotonin in the brain gut axis has a role to play. Important in secretory and peristaltic reflexes, signals to the vagus nerve to signal to the brain in the generation of nausea. This is how somebody hears the bad news and feels nauseated and has to go to the restroom. Enterochromaffin-like cells or ECL, they secrete histamine, okay, and they secrete it locally. This is a paracrine secretion, and it stimulates the parietal cells to secrete more HCl. So the enterochromaffin-like cells, they will trigger, they will secrete the histamine, and it will cause it will stimulate the parietal cells to produce more HCl. So what are the cells that produce HCl? Parietal cells. How do they produce it? I already showed you by splitting the bicarbonate, the carbonic acid into bicarbonate in exchange for chloride. They produce or generate more HCl. Why do they produce HCl? Because that HCl is going to work on pepsinogen, which is a proenzyme, which is a sleepy inactive enzyme. Once HCl works on it, it's going to get activated. Why do we need it? Because an active pepsin can work on the protein and break it down into amino acids. Digestion would be completed. Now the question is, what other cells can also instigate parietal cells to produce more and more HCl? One would be gastrin hormone, and the other one would be also the ECL or the enterochromaffin-like cells that would cause the parietal cells to secrete more HCl. They secrete histamine, and they secrete it locally like a paracrine secretion, and they cause the parietal cells to produce more HCl. So over here, parietal cell has receptors. Parietal cell has receptors. Why does it has receptors? It has receptors because it is responding to gastrin, because it's responding to acetylcholine, because it's responding to histamine. So because that's why they tell you, don't take so much of anxiety, don't take so much of stress. Why? Because if you are constantly in stress and worry and anxiety, you are going to activate the sympathetic nervous system more and more, fight or flight mechanism. It's going to produce more and more kind of your acetylcholine will be activated, your neurotransmitters are going to be activated, and these neurotransmitters will cause your G cells and your nerve endings will be activated, and they will cause these paracrine secretion of the ECL to produce more histamine, and all of these can bind to the receptors of the parietal cell and force it to produce a lot of HCl. If you produce a lot of HCl, you are producing a lot of gastric juice. Finally, if you don't eat food at the right time, this acid is going to eat away the wall of your stomach and cause big peptic ulcers or gastric ulcers. That's why they say the brain gut axis, or the stomach and the brain connection, or emotions, our tempers, or our how we process. Don't take stress, don't take anxiety, don't be worried if he tells you something bad, don't be upset. It's easy to say, it's difficult to live it out, because we all go through stress. But the sooner we try to, cope up with it, or divert our mind into something that is productive, or think about something good things, or, channelize our energy, or spend time with people who see our strength, or who would understand us, that would relax the mind. That's the mind and the body connection. So the more stress we take, the more fight and flight mechanism we land into, it harms our body eventually, because it's going to cause more acidity inside our body, because these nerves are going to work through these hormones and trigger the parietal cell to produce more and more HCl. And if there's not enough food inside, the HCl is going to eat away the walls or the of the gastric lining and cause ulcers. So coming back to the topic over here, the parietal cell, how it generates more amount of HCl. So water plus carbon dioxide in the presence of carbohydrate enzyme is going to produce this carbonic acid that's going to split into bicarbonate and H plus. Okay, and over here the exchange happens where bicarbonate is exchanged for chloride. So now chloride enters, and here's use of ATP energy, and with that the hydrogen ions and the chloride combine to form HCl. That's how the parietal cell produces a lot of HCl. Look at the receptors. It has a receptor for acetylcholine that's connected to your mind. It has a connection with histamine that is produced. It's a connection with gastrin hormone. All of these are the brain gut axis connected to your mind to a great extent. And then that triggers more production of HCl. And if you're not careful, it's going to cause ulcerations or deep wounds in the wall of the stomach if you don't eat food. And the acid is produced in such a great amount, there's no food to act on it. There's no protein on which it can act. It's going to attack the wall of the stomach itself. So chief cells, as you know, they produce pepsinogen, that is inactive. Okay, it's a proenzyme, it's a zymogen, and it's inactive. That is converted into active pepsin. And who does it? HCl. HCl is secreted by whom? The parietal cell. So this is the location of the HCl producing parietal cell. Pylori glands, they contain D cells that secrete somatostatin, and it inhibits or prevents gastrin secretion. You can't have gastrin secretion flowing, overflowing. There has to be a stopping. There has to be a limitation, and that's done by somatostatin secreted by D cells of the pylori gland. So this is important. Question for you over here. Cells lining fundic part of the stomach, they secrete ghrelin, and that initiates hunger before meals. Ghrelin is antagonistic or opposite in function. Antagonistic means opposite to leptin. Leptin is derived from the fat tissue, and it provides satiety. Satiety means fulfillment or satisfaction. You took one slice of pizza and you are satisfied. You don't need a second slice of pizza. That satisfaction, a fulfillment, comes through leptin secretion. Now, but it is derived from fat tissue. So if you are eating too much and becoming obese, then the fat incorrectly starts functioning incorrectly, misbehaving in such a way that the leptin will never be secreted in such a manner that you don't reach the satisfaction level. You don't reach the fulfillment or the satiety. So one pizza, you take the second pizza and the third pizza and end up eating 8 pizzas slices, and then you say, oh, I gained a lot of weight. So leptin, blame the leptin. Why? Because too much of fat cells will completely confuse leptin, the way leptin should behave in the path. So the satisfaction is never reached. Ovastatin, that decreases the appetite and inhibits the thirst. So all of these hormones are important. Who does what function? Now let's look at the stomach histology. We're running short of time. So muscular is externa. It has 3 layers: the inner oblique, the circular layer, and the longitudinal layer. These are the 3 layers. That allows the stomach to churn the food or mix the food and form the chyme. Chyme is food plus all the enzymes and the gastric juice mixed up together. The milky substance forms when food mixes with the gastric juice, and then it moves down. So the propulsion system, how the food passes through the stomach. We have the pyloric valve over here, as I had shown you. And over here, the pyloric valve, as you can see, is closed. So the food is stuck there. Then propulsion, peristaltic movement happens in the forward direction. Grinding, vigorous peristalsis or mixing happens right here in the peristaltic movement, and then the valve will slightly open over here, and you can see the food will come down. Retropulsion, the pyloric end of the stomach acts as a pump that delivers small amounts of chyme into the duodenum. Small amount comes into the duodenum, forcing most of its content material backward into the stomach. So it one arrow, most of it goes in this backward direction. Little bit comes in the forward direction when it slightly opens, and then it closes, and then slightly opens again, little bit forward. So little by little, the food moves towards the small intestine. That's the movement. Gastric juice, 2 to 3 liters per day. And importance of HCl, it activates the pepsinogen, that is a propepsin or zymogen. It is inactive, it activates it. So we have pepsinogen converted into pepsin by HCl. This is inactive, secreted by chief cells, and it converts into active pepsin. HCl is secreted by parietal cells. Pepsinogen is secreted by chief cells. This active pepsin in the next step is going to act on protein and break it down into simple amino acid, complex protein into simple amino acid, and that's chemical digestion. Control of GI tract. So we talked about gastrin, that's secreted by enteroendocrine G cells. It will cause the parietal to produce more HCl. We already did this. Histamine, that is paractin local secretion. Enterochromaffin-like cells will secrete, and they will cause it parietal to produce more HCl. Secretin, secreted by the duodenum. We'll read about it a little later when we talk about intestine. Then it reduces. Now this is important. The secretin reduces the acid secretion. So question for you over here. Gastrin increases the secretion. Histamine increases. Secretin decreases the acid secretion. And it stimulates bicarbonate buffer production by the pancreas. Cholecystokinin, CCK, that's secreted by the duodenum of the small intestine. It causes release of the digestive enzyme from pancreas and gallbladder for breakdown of protein and fat. So cholecystokinin, that is secreted by the intestine, but it goes to the pancreas and the gallbladder and causes them to secrete the enzymes. So cholecystokinin has a very important function. Secretin, secreted by the duodenum, it reduces the acid secretion. So name a hormone that increases acid secretion, gastrin. Name a hormone that reduces gastric secretion or HCl secretion, that is secretin. name a hormone that is secreted by the intestine that goes to the gallbladder and the pancreas and causes them to secrete their enzymes, and that is cholecystokinin. Nervous system. So the nerve control of the GI tract in this region would be the parasympathetic vagus that causes increase of the acid production, and the enterogastric reflex. It helps to shut down the acid production that involves the sympathetic and the CCK and secretin. So question for you. Name the nervous system that increases the acid production. That is parasympathetic nerve or the vagus nerve. Name the nervous system that would reduce or help to shut down or reduce the acid production. That would be sympathetic, CCK hormone, secretin hormone. Both of these secretin and CCK are produced by the duodenum of the small intestine. So this is important page. Any questions? So if you don't have any questions, we'll proceed down. Control of the stomach. So the control of the stomach is divided into cephalic phase. Cephalic means the head part. Cephalic phase, I mean, not this head, but the initial phase of the control. Then the gastric phase and the intestinal phase. So the first phase where gastric acid secretion begins, when you see the food, when you smell the food, when you think about food, how does it work? The hypothalamus in the brain would pass the information to the medulla. The sensory information has come through your sense organs of seeing visually the food, or somebody talked about food, or you watched something about food. So that gets information triggered through the hypothalamus, medulla, comes through the cranial nerve, 10th vagus nerve, and then the gastric activity begins. Okay, the gastric activity begins. So parasympathetic mechanism would be activated and your HCl production would begin. Gastric phase, acid secretion due to the presence of food in stomach, where this is where the actual food you have eaten comes into your stomach, and it causes stomach to stretch, also due to the presence of amino acid in stomach. pH will drop because it will be an acidic medium due to the presence of food, and vagus nerve and pituitary are all stimulated. Gastrin is released. All of this will lead to secretion of HCl. It will cause the parietal cells to produce more HCl, and acetylcholine, histamine, gastrin, all will increase gastric secretion. So this is a question for you, important question. Which all components increase gastric secretion? In the cephalic phase, when you look at the food, when you think about food, when you watch somebody eating, when you hear somebody talking about food, through the sense organs, hypothalamus activates medulla, medulla activates vagus nerve activates the parietal cells and causes gastric activity inside your body. In the gastric phase, the actual food enters, and then the gastrin is released, the histamine, acetylcholine, all of these have receptors in the parietal cell. They're received by the parietal cell, they trigger it, they cause the splitting of the formation of bicarbonate. Bicarbonate is exchanged with chloride. You produce a lot of HCl. Everything that we have already discussed so far. You produce a lot of gastric secretion, a lot of HCl. The last one is intestinal phase. The acidic kit, now it is mixed up with all the enzymes, enters the duodenum, that is the small intestine. pH is less than 2. It's very, very bitter. It's very highly acidic. And gastric secretions now are inhibited. Now that the food has passed from the stomach, entering into the duodenum of the intestine, the gastric secretions are no longer required. HCl is no longer required. So now it would be inhibited. Who would do the inhibition? Sympathetic, the CCK, and the secretin hormone. Secretin, CCK, cholecystokinin, and the sympathetic pathway, they would stop the gastric secretion. So low pH carries into via the vagus nerve to the medulla, reduces the gastric secretion. Secretin, cholecystokin produced by duodenum, they decrease the gastric secretion. So whatever I explained just now, diagrammatically, it has been depicted over here. You can go over the diagram. Cephalic phase, stage 1, gastric phase, and then intestinal phase. How the food first comes through the gastric phase, passes down, and then enters into the intestine. So it shows in the stimulation, all those who stimulate or cause the acid production, those are shown in red. And you can see all the blue ones that are inhibitory would be shown in blue. CCK is blue. Then secretin is blue, GIP is blue. All of these are inhibitory, stopping the flow of gastric juice because now the food, the chyme has entered into the small intestine. At the initial stage, when you have sight, smell, taste, thought of the food, all of these, then you will find that these cells are activated to produce, parietal cells will produce HCl. The G cells will cause the parietal cells to produce more and more gastrin. So this is a step by step, phase by phase. It shows how pepsinogen is secreted, how the HCl will be secreted, acts on the pepsinogen to convert it into pepsin. These are the stretch receptors and the chemoreceptors that will act via the myenteric plexuses. This is the enteric nervous system that regulate these receptors. So here it shows whatever I explained to you, the same steps. Chyme entering the duodenum causes CCK and secretin. They will shut down the gastric juice production. And CCK will induce the secretion of pancreatic juice. So CCK, cholecystokinin, that is secreted by the duodenum of the small intestine, that will go and act on the pancreas to cause the pancreatic juice secretion. And secretin will cause the secretion of bicarbonate rich pancreatic juice. So both of the CCK and secretin, they're both secreted by the duodenum of SI. SI means small intestine. And they both go and act on the pancreas, which is the accessory digestive organ. And they will, accessory GI tract organ, and they will act on it to cause it to secrete the pancreatic juice so that the pancreatic juice will enter and help in the remaining part of the digestion. So this is how it works. So this is the pancreas. This is the, right here, this is the duodenum. Okay. So the duodenum, the CCK and the secretin are here. Okay. So they are going to act on the pancreas and stimulate the pancreas. Look at the arrow. They're going to come and act on the pancreas so that pancreas will be forced to secrete the pancreatic juice, and that pancreatic juice will help in the digestion of the remaining part of the food, which is in the small intestine. So digestion, mechanical, we have done. Chewing, chemical. It involves enzymes and the process of hydrolysis or breaking the bonds. So in case of carbohydrate, when the salivary amylase acts on it, the glycosidic bonds between the sugar molecules, that is broken. So breaking bonds in food we eat to make them smaller. And by the time food reaches the jejunum of the small intestine, digestion should be completed. So when we reach up to jejunum, digestion should be completed. Enzymes are proteins specific for one reaction and one reaction, like a lock and key model. Okay. And it can be used over and over again. It catalyzes or speeds up the reaction. It works in a specific pH and a specific temperature. These are the basic general characteristics of enzymes. You must definitely be aware of it, about enzyme characters. So next we move to absorption. So broken down food molecules are absorbed. So now we have digestion completed. For example, let us look at protein. So we have pepsinogen that is inactive, and that's converted into active pepsin. And who did it? HCl did it. This was secreted by chief cells. This was secreted by parietal cells. So we have converted it into active pepsin. Active pepsin now works on the protein and it converts it into amino acid. So this is active pepsin. It acts on protein complex and breaks it into simple amino acid. All of this digestion should be completed by the time it reaches up to the jejunum. Passing of the, now defecation is the passing of the undigested food, cellulose, or some of the plant products which are undigested. So these have to be removed out of the toxic substances. These involve the process called defecation, passing of the undigested food, or the production of the poop, uh, or the feces. Muscles in the rectum, they will stretch, sensation of having to running to go to the restroom. So two muscles are involved. What are the two muscles? Internal anal sphincter, that's a smooth muscle, sensation of defecation, and then external anal sphincter, that's a skeletal muscle, voluntary control. So here in the rectum, and this is the anus, okay, this is the rectum, we have two of these muscles sitting here. We have two muscles sitting here, okay. So the first one is the internal anal sphincter, and this is a smooth muscle. It's a smooth muscle. That means when the poop comes down, we do not get the feeling yet, because it's smooth muscle, it will involuntarily pass down. We get the feeling of going to the restroom when it comes at this level, where it is a skeletal muscle, and we control when we are going to look for a restroom for the definite place. So that is under skeletal muscle control. So a question for you would be, out of these two muscles, which one is smooth muscle and which one is skeletal muscle, and why? The internal anal sphincter is smooth muscle, we have no control over it, whereas the external anal sphincter is skeletal muscle, and we have control, voluntary control of the defecation. So now here it will show you the steps, how the defecation process happens step by step. Look at number one. Distension or the stretch of the rectal walls due to movement of the feces will happen. So first of all, when the poop is coming over here from the colon into the rectum, we have stretch receptors. These are stretch receptors. These are stretch receptors, okay. So the wall of the rectum is going to swell up or expand or extend because the poop enters, and the stretch receptors, they will provide sensory or afferent. Look at the arrow, look at the arrow, look at these arrows, they are going in the forward direction. They will provide the information, sensory or afferent or onward or going towards the spinal, this is the spinal cord, to the sacral region between S2 and S4. They will provide the information to the spinal cord that we are stretching because there is poop that needs to be removed or that needs to be defecated. So the receptors, they transmit signals along afferent. Afferent is sensory, going towards the brain or the spinal cord. So in this case, towards the spinal cord neurons. So it carries the message, the information. Who sends it? Stretch receptors present on the wall of the rectum. They provide the information. Now the spinal cord receives it, and then there is a defecation reflex that gets activated. So when that is activated, it is in this reflex is initiated, in which parasympathetic motor means you want some action to be done, efferent fibers stimulate the contraction of the rectal walls, and first thing they do is they contract the rectal wall. So now this information comes like this, okay, it comes like this to the rectum wall, and it causes contraction of the rectal wall. The arrow is downward, okay, and it through the motor or the efferent nerve, now it comes, it causes the contraction of the rectal walls and relaxation of the internal anal sphincter. Contraction of the wall, but relaxation of the internal anal sphincter. Please do not get confused between which one is contracted and which one is relaxed. The anal rectal wall is contracted, the anal sphincter is relaxed. When it is relaxed, that means the involuntary motor nerve parasympathetic division acts, and it causes the internal anal sphincter to relax. When it relaxes, you get the urge of the poop because now it is going to come down. Okay. Then if it is convenient, if you can find a restroom toilet somewhere nearby, then if it is convenient to defecate, then the voluntary motor neurons are inhibited and allowing the external anal sphincter to relax. So next stage is the external anal sphincter is now going to relax, and so that the feces may pass. This is called as the defecation reflex. Any questions? 12:09. So we have to rush through the remaining slides. Let me see. So small intestine is almost approximately 23 feet, not exactly 23, and it's divided into duodenum, jejunum, and ileum. Let's look at, first of all, the duodenum. It extends behind the posterior parietal peritoneum. It is a C-shaped structure. This part of the duodenum has an adventitia. That means, as I told you, it is retroperitoneal, and we'll look through its histology and its digestion. This is the wall and the cells that are present. Okay, so it has simple columnar like just the others, and the exceptional characteristics it's that it has the mucosa is folded into villi or microvilli. What is the difference between villi and microvilli? So villi is like finger-like projections, which allows for more surface area for absorption. Microvilli is like hair-like structure. That is a brush border, a hair-like structure, and that's called microvilli. And on top, to increase the surface area of absorption. And it also has plicae circularis, that is folds of both the mucosa and submucosa of the intestine, and even more surface area for absorption. So question for you would be, what is a villus? What is a microvillus? What is plica circularis? This is folds that will help in more expansion of the surface area for absorption. Goblet cells, what's their function? Makes more mucus to neutralize acidity of the stomach. So goblet cells produce mucus. Mucus will neutralize or defeat the acidic content. Paneth cells, they make lysosome. What is their function? Antibacterial, to prevent the infection. Enteroendocrine cells, they secrete CCK and secretin. We already talked about cholecystokinin and secretin. Both are secreted by the duodenum. They go to the pancreas. I showed you how they go to the pancreas, and then they cause the pancreas to secrete the pancreatic juices like the pancreatic amylase or the pancreatic lipase. So CCK and secretin, and both of them stop acid work of parietal cells. They inhibit acid secretion or prevent acid secretion. Lacteals in the lamina propria. This is important. It helps in the absorption of fat. I will explain about how the fat gets into the lacteals in the villi. So the muscle layers, they have circular folds. These are the finger-like projections or villi. This is the lumen or the internal cavity through which the chyme comes, and the vein carrying blood to the hepatic portal vessel. So from the small intestine, the veins are connected, the artery and the veins are connected to the hepatic artery and the hepatic portal vein of the liver. The liver is also called as the hepar. That's why the term is hepatic. Now, if you look at the wall of the intestine, just like the stomach, it has a mucus, submucosa, muscular layer, and the serosa, which is the topmost layer. It has additional parts called villi, intestinal glands. They're present in the crypts of Lieberkühn, and this is epithelial. They have, if you look at one of the intestinal glands, you can see the Paneth cell that secretes lysosome, that is antibacterial. If you look at the tip of a villus, it consists of lots of goblet cells, these white structures. Even if under microscope, when you're doing labs, you can look at the goblet cells. These are the goblet cells, white balloon-like structure, and the villus, and these are the lacteals. The lacteals absorb the fat, the long chain fatty acid. They absorb the fat and they push it into the subclavian vein. So if you look at the internal structure of a villus, you can find the red one is the artery, the blue one is the vein, and the green one is the lymphatic channel, and the yellow one is the nerve. So duodenum history, submucosa, the submucosa contains glands, which are called as Brunner's glands in this case, and they produce bicarbonate rich mucus that will help in, wherever you see mucus, you will know, oops, Wherever you see mucus, you'll see that it will neutralize the stomach acid. Mucus also helps to protect the epithelium. And it has muscularis layer, adventitia. Well, the whole duodenum is not retroperitoneal. I've told you the first part is intra. Remaining part has the adventitia. That's why remaining part is retro. The top part is serous. Things that enter the duodenum. Acidic chyme enters from stomach, bile comes from the liver through the common bile duct, and pancreatic juice enters from the pancreatic duct. So if you look at the duodenum is a very strategically very important location, and it has multiple functions. It's very, very crucial role to play because it not only assists in digestion, it also receives the enzymes that come from the liver and the pancreas. So it receives the bile from the liver, it receives the pancreatic juices from the pancreas, and it also receives the food or the chyme from the stomach. So three things enter. The chyme comes from the stomach, the bile comes from the liver, the pancreatic juice comes from the pancreas. So three things enter into the duodenum. Lipase comes from the pancreatic lipase comes from the pancreas, and it causes the fat digestion. Amylase comes from the pancreatic amylase comes from the pancreas, and it does the carbohydrate digestion. And proteins, they have zymogens activated that would act on the protein to convert it into amino acids. Now, how does it work? Let's see. So in the wall of the duodenum, in the epithelial cells of the duodenum, sitting right here, there is enteropeptidase enzyme. It is also called as enterokinase or enteropeptidase. This one is going to act on inactive trypsinogen and convert it into active trypsin. That's the first step. The first step is to convert the inactive trypsinogen into active trypsin. Who does that? Enterokinase or enteropeptidase does it. Where is it located? It's located in the wall of the duodenum. Once it converts the inactive trypsinogen into active trypsin, the next step is that this active trypsin in the second step is going to act on the chymotrypsinogen inactive and convert it into active chymotrypsin. It's also, the trypsin is also going to act on inactive procarboxypeptidase and convert it into active carboxypeptidase. Your question is, this guy was present in the wall of the duodenum of the small intestine. We can understand that. Where did these three come from? Where did these three come from? They came, look at the source, they came from the pancreas. So if you remember, I told you CCK and secretin. These two hormones were present in the duodenum. They went to the pancreas and they caused the pancreas to release these enzymes. But unfortunately, these three enzymes were in the inactive state. They are called as zymogens inactivated. If they are inactive, they can't do any digestion. So they have to be converted into their active state. Like going back to stomach, like we converted inactive pepsinogen into active pepsin thanks to HCl in the stomach. Now, in the intestine, these inactive enzymes have to be converted into active trypsin, active chymotrypsin, active carboxypeptidase. Who is going to do that? This enterokinase or enteropeptidase, which is sitting in the wall of the duodenum, is going to do it. So, again, we are going to go over it because this part is important. First step, we have CCK and secretin. Okay, these are hormones present in the wall of the duodenum. These hormones, they resist acidity. Forget about it. First thing, these two go to the pancreas and they cause the pancreas to release enzymes into the duodenum. Step two, once the pancreas releases these enzymes, we realize that these three enzymes are proenzymes called zymogens, and they are inactive enzymes. They are good for nothing unless we activate them. The third step is Here we have enteropeptidase, a membrane-bound enteropeptidase, also called as enterokinase, that's going to come and convert trypsinogen into active trypsin. The next step, its job is done. Enterokinase job is done. In the next step, active trypsin is going to act on inactive chymotrypsin and convert it into active chymotrypsin. And the next step, again, trypsin is going to act on inactive pro-carboxypeptidase and convert it into active carboxypeptidase. All of these three, trypsin, chymotrypsin, carboxypeptidase, are going to work on protein and break it down into amino acid, and that would complete protein digestion. Any questions? Is it clear? You need some response. Is it clear? Or you need more clarification? Or what are your questions? Or please give a response, any feedback. Yeah, am I going like too fast, or do you want it to be slowed down or to be repeated? You can chat your responses. Please provide your feedback so that we can proceed to the next slide. Okay, thank you. Thank you for the feedback. So, okay, thank you. I too need some encouragement, right? Okay, so okay, thank you everyone. So let's go to the next slide, that is histology diagram. Clear. Yes, I believe in diagram 100%. Without diagram, I would not be able to get the satisfaction that I explained it to you properly, because it's a lot of information, and the diagram, it really helps. And I also want you to learn by drawing diagrams, because it's not just that I teach and you understand, but that when you study the slides or when you study the lecture notes, that you would draw simultaneously and trying to code a diagram with your own diagram, with the understanding of the topic or your textbook that you follow, or the lecture notes. That would keep a visual kind of an image on your mind, and when you go for the exam, it will make things very, very simple. The diagram will appear in front of you more than the text or the memory of those sentences. So histology of jejunum. So again, this consists of mucosa, submucosa, muscularis externa, and serosa. Look how all of these walls of the alimentary canal are more or less similar. The exceptional things you have to remember. For example, stomach wall. It has additional layer of circular muscles, longitudinal muscles, and oblique muscles. More muscles are present in the stomach wall. That's the exception, because you have more food content, more expansion, more folding of the rugae, and more holding the food for longer time, more churning. So you need more muscle strength. That's why it justifies that muscle layer of stomach has 3 layers. In case of intestine, you need more absorption. Digestion is completed, churning is completed, so you need more absorption. So you see villus or finger-like projection, villi or circularis, plica circularis, all of these additional parts in the intestine, which is not there in the stomach. And then you find that in duodenum, we talked about Brunner's gland. Brunner's gland is not there in jejunum, because our digestion mostly happened in the duodenum. So we are done with Brunner's gland. You will not see them in jejunum. So question for you here, it would be, do you have Brunner's gland in small intestine? Yes, we have, but only in the duodenum. We do not have Brunner's gland in the jejunum. Why? Because our maximum absorption is finishing off in the duodenum. Coming to the jejunum, we don't have requirement of so much of digestion or the remnant part of the absorption. So that is why you won't find Brunner's gland in that part. Well, maximum absorption of water will take place in the jejunum. Submucosa and mucosa thrown into larger folds called plica circularis, that is there. It slows the food down and allows for maximum absorption of food from mucosal villi. Then muscular is externa, remains the same, and then serosa. We have larger villi than in the duodenum. This is an exception. We have larger villi. Chemical digestion in the jejunum, we have brush border enzymes. So you would need to know the names of these enzymes. We have disaccharidases, lactase, sucrase, and maltase for sugar absorption, and then we have dipeptidases that would work on the protein. So you need to remember the name of these brush border. I explained to you, I think brush border is these hair-like processes, villi are the finger-like projections. So site of absorption would be jejunum, and modification of the small intestine to increase the surface area. That's why we have larger villi over here. Maximum water absorption would happen in the jejunum. Then comes the ileum, the last part of the small intestine. It also has mucosa, submucosa, muscularis externa, and serosa. Again, note over here, no Brunner's gland as seen in duodenum. It again has simple columnar, villi and microvilli still present, but much less because we are done with the absorption. We are slowly reducing the absorption. Lamina propria contains Peyer's patches. So now an exception. What is Peyer's patches? It has aggregated lymphoid nodules that will provide protection against bacterial infection. These are the Peyer's patches that you can see under the microscopic structure. These are lymphoid nodules and will prevent bacterial infection. So as you go stomach to small intestine, each part will present some unique characteristics. Rugae present in stomach. They have sphincters over there. Note it down. You go to duodenum. Duodenum, you will find that it has a very strong connection with the liver and the pancreas, receiving enzymes from them, receiving bile from the liver. And when you go to the, you have villus. You don't find villi in the stomach. You have villus in the small intestine. So as you go through each structure, note down the exceptions that are present. In this case, Peyer's patches are present. What are they? They are lymphoid nodules. What do? They fight against bacterial infection. Brunner's gland is present in duodenum. Brunner's gland is not there in jejunum and ileum. So note it down. Now, Peyer's patches are immune cells, lymphocytes, and macrophages. They're aggregates from lamina propria connective tissue, and they extend is more into this layer. It also has muscle and serosa. Movements in the small intestine, I think we did this one. This slide is a repetition. Anyway, so peristalsis is a unidirectional forward moving propulsion, pushing the food like through a toothpaste, and segmentation is the churning in the back and forth direction, and it helps it forward and backward direction and slowly propels the food forward. So next we go to the last intestine. We're going to study about the anatomy, histology, formation of feces, and how feces are formed, and then the absorption of water. So if you look at the large intestine, what are the parts? It begins with the cecum or the appendix. Okay, so here's the cecum, the appendix, a vestigial organ that we do not use, and then it goes up as the ascending one. Then comes the transverse or the horizontal part, colon, then the descending colon, and then the sigmoid. This part is like S-shaped sigmoid, and then rectum, and then anus. So cecum, appendix, goes up, ascending, transverse or horizontal, goes down as descending, S-shaped sigmoid, rectum, and anus. Now, you will find in the large intestine, these rostrum or kind of a pouches, okay, maximum storage for the fecal matter, and you'll find a muscle called as tinea coli that kind of forms these patches. This tinea coli forms these patches, pouches, or like small bags, allowing maximum expansion. If you look at the wall, again, it is simple columnar, no villi, we don't need any absorption, but some amount of water absorption still happens. A lot of goblet cells, a lot of mucus production is there, and we don't need desiccation or drying of the large intestine, so a lot of mucus production. Lymph nodes to fight against bacterial infection, because if the large intestine has fecal matter, feces, lot of bacteria, so need lot of lymph nodes. Then submucosa, muscularis mucosa, again circular muscles, longitudinal muscles, no oblique muscles. And then it is partially contracted into haustra or pouches or puckers because of the tinea coli muscle. Serosa part is okay. Now the large intestine, as I had told you, part of it is intraperitoneal, part is retroperitoneal. So the serosa part, which is intraperitoneal within the peritoneal cavity, that would be the transverse colon and the sigmoid colon. Whereas the ascending, the descending, and the descending rectum, this is all adventitia attached to the abdominal wall through fibrous connective tissue collagen. And this is retroperitoneal. This is within the peritoneal cavity. So intra and retro. So questions may come to you, which part is intra and which is retro. Water absorption, 90% of water absorption finishes off the jejunum. Only additional 9% occurs in the large intestine. That's why we don't have villi. Formation of feces, undigested food, what all appears in the feces, fecal matter, that is bicarbonate secreted in exchange for uptake of NaCl. So you remember the NaCl that we had exchanged to produce the acidic medium. That is why we had exchanged it with bicarbonate, right? So that comes out. And then bacteria, we provide them a good place to stay, the good bacteria, and they provide us with vitamin K and vitamin B. So the good bacteria help in extracting the vitamin K and B from the food, and eventually this helps in clotting and metabolism and immune function. We have epithelial cells also. Epithelial lining is constantly renewed, it shreds off, okay, and because of abrasion, and old cells are sloughed off and they become part of the feces. So if you go for a occult fecal test, you should see some epithelial cells over there, some good bacteria. It should not have too many pus cells, it should not have blood. Rectum, so histology and the defecation reflex. We already completed the defecation reflex, so I will not go back into it. I've already explained to you. And just remember that the rectal region is connected to the sacral region through the pelvic nerve, that is parasympathetic, and the pudendal nerve. I had shown you how when the fecal matter comes, the receptors are activated, then it through the nervous system, it activates the sensory nerve afferent, it informs the spinal cord, and then the reflex, defecation reflex is triggered. So then the efferent or the motor nerves gets activated, it relaxes the internal sphincter, and the feces comes down, pulls down to the anus, and then through the skeletal muscle control, we regulate the external anal sphincter and decide when we can go to the restroom. This is a table that shows you the type of epithelium in each part. In the stomach, the duodenum, jejunum, colon, appendix, anus, the type of epithelium, the type of cells that are present, and exceptional features present in each of them. This is a very important table for comparison. So now we come to the chemical part of the chemical digestion. Remember, carbohydrates contain carbon, hydrogen, oxygen in the ratio 1:2:1. We know that complex carbohydrates have to be broken down into monosaccharides, that is glucose, fructose, galactose. This is all school level. Definitely in your high school, you must have studied it. Disaccharides, two simple sugars that are condensed through dehydration process to form sucrose, maltose, and lactose. And polysaccharides are always definitely glycogen and starch. Cellulose, our body cannot break. Let's look at the chemical digestion. We generally take in a lot of starch and the polysaccharide that consists of several glucose molecules. So whatever we are eating has to be broken down into simple sugar. So basically we take the carbohydrate and that is broken into glucose. Whatever protein we take is broken into amino acids through chemical digestion. Whatever fat we take is broken into fatty acid plus glycerol. So digestion, digestion, and digestion. Remember hydrolysis? So we use water to break the bonds. For example, for a carbohydrate, we use water, the process of hydrolysis, to break down the glycosidic linkages or bonds between the sugar molecules. So our body makes plenty of water. So we need water hydrolysis. So drink your water, because if you drink water, it will help in digestion process. Saliva and the gastric juice, pancreatic juice, all of them, they contain water. So enzymes are proteins specific for one reaction only, catalyzes specific pH and temperature. I think we had done this slide. Now, let's look at the digestion. Polysaccharides converted into disaccharides. I mean, converted means broken down, then disaccharides broken into monosaccharides, and then this glucose is absorbed into the blood. It begins in the mouth. We have done the salivary amylase. Amylase breaks down the starch or the polysaccharide into disaccharide. So we have in the mouth carbohydrates breaking down into disaccharides and glucose thanks to salivary amylase enzyme. In the stomach, not too much of carbohydrate breakdown here, unless amylase surface is hidden within the food bolus. In the stomach, it's very acidic. pH is 2, and the amylase pH is 6.5. Next, in the duodenum, pancreatic amylase enters. Who causes it to enter? We have already studied this. CCK and secretin. They both act on the pancreas, causing the pancreatic amylase secretion, and that enters. Then jejunum, it has brush border, simple columnar. Give me 1 second. I have a bug over here. Okay, sorry. Jejunum, brush border, simple columnar epithelial lining. Cells have surface extensions called microvilli, and the enzymes that exist here, lactase, sucrase, and maltase, disaccharides. So in the jejunum, these enzymes have entered. These enzymes are there which will convert the carbohydrate into simpler polysaccharides converted into disaccharides. That means lactase is going to act on lactose, sucrase will act on sucrose, maltase is going to act on maltose. So over here, as you can see, the pancreatic amylase is releasing these enzymes right over here. The CCK and the secretin have acted on the pancreas to cause it to release the pancreatic amylase. So as the starch enters, you can find maltose and oligosaccharides. The digestion process continues, and this oligosaccharide is converted into simple glucose. Who does it? Maltase, dextrinase, glucoamylase. From where did this come? They all came from pancreas. They all came from pancreatic amylase, and all of this came here. They broke down. These enzymes are acting over here, involved over here, converting the complex carbohydrate into simple glucose, and so that absorption can happen. Why did the enzyme come from here? Because we had CCK and secretin secreted by the duodenum going into the pancreas, causing it to secrete or release the pancreatic juice that contained all of these enzymes that converts the carbohydrate complex into simple glucose that can be absorbed. So carbohydrate digestion and absorption in the small intestine. Now, during this digestion and absorption process, something very interesting also happens. You can see that the polysaccharide, with the help of enzymes, breaks down into oligosaccharides and disaccharides. Okay, oligosaccharides, they have multiple, like 8 components of saccharin sugars, and poly is multiple. It's breaking down into 2 sugar, and then ultimately we need glucose, simple sugar, single sugar. We need glucose that can be absorbed into the blood. So now the breaking process has been completed by the pancreatic juice or the enzymes, maltase, lactase. All of these have broken down the complex carbohydrate into simple glucose. But once the digestion is done, how do we take it to the blood? How are we going to take it to the blood? That work was completed by amylase. Breaking down is done. We got the simple glucose out of these oligosaccharides, disaccharides, and polysaccharides. But how are we going to take the simple glucose and put it into the blood from the digestive tract or from the GI tract? We do that depending upon the sodium-potassium pump. Now, the sodium-potassium pump, it kind of creates a gradient where sodium and potassium move in and out, causing a gradient, and because of that gradient or concentration difference, some amount of ATP energy is definitely used for that. Because of this exchange, glucose that was broken down, it kind of jumps on or latches on to the sodium, and as the sodium moves out, this glucose tries to come out with it. So sodium glucose form a kind of a co-transporter. Sodium, in exchange for potassium, this sodium has entered and it kind of allows the glucose to latch onto it like a co-transporter. I mean, you are going in a vehicle somewhere and I ask you for give me just I need a car drop somewhere, so you pick me up from a location and drop me at the college. So you are being a helpful friend to me. So sodium right now is a co-transporter helping the glucose to come navigate and come down into the bloodstream. That's part one. In the second part, you'll find that part of this, like fructose, the end product of carbohydrate digestion, will need a facilitated diffusion mechanism. Somebody is going to help it. Facilitated diffusion, some transport proteins will come to the rescue. This light green is a transport protein. It will help in its rescue and it will bring the fructose down into the blood. So we have two different mechanisms by which, once the carbohydrate is broken down, the complex one, that is polysaccharide or oligosaccharide, is broken down into glucose or monosaccharide by pancreatic amylase. Once that is done, the simple glucose is now assisted down from the lumen of the intestine where the digestion happened, from there into the blood in the capillary, to be absorbed. How is this absorption happening? Because we are depending on sodium potassium pump, which helps the sodium to act as a glucose co-transporter, helping it to carry the glucose up to the blood. That's step one. The step two is fructose is also an end product of carbohydrate digestion. It takes the help of facilitated diffusion by latching on to a protein transport mechanism and making its way up to the absorption. So that is how after the digestion, these carbohydrate end products make their way into the blood. Now, talking about this part, we also need to understand how the fat gets into the lacteal. So in the villi, you can find that there is a green channel that you can see over here. This green part is the lymphatic part, the lymphatic portion. We call that as lacteal. Okay. So now when fat breaks down, it breaks down into fatty acid and glycerol. Okay, fatty acid and glycerol. This fatty acid has long chain fatty acid, it has short chain fatty acid. The short chain fatty acid will directly go and get absorbed into the blood. But the long chain fatty acid, they will enter into each of this villi, and in the villi, you will find this structure called lacteal. The long chain fatty acid cannot directly get absorbed into the blood. They will get into the lacteal, and from the lacteal, they will be taken via the subclavian duct, and they will enter into the lymphatic system. We'll talk about them later on when we go to the lymphatic system. But for now, you understand this much: the end product of fat digestion is fatty acid and glycerol. The short fatty acid chains will get absorbed directly into the blood. The long fatty acid chain will enter into each villus, and these villus have lacteals into which it enters, and from the lacteal, it will go into the subclavian duct. That's the end part of fat digestion. So in a nutshell, for carbohydrate digestion, from complex carbohydrate or starch to monosaccharide, what are the enzymes we use? We use salivary amylase in the mouth. We use pancreatic amylase coming from the pancreas. We use these brush border enzymes, lactase, sucrase, maltase. All of these are useful in breaking down carbohydrate into glucose and through the process of hydrolysis. Come to protein digestion. Protein digestion begins in the stomach. This point is important, where it begins, okay? And it is broken down into amino acids by hydrolysis, same process. But no protein digestion happens ever in the mouth. Most occurs in the duodenum, where CCK and secretin will go to the pancreas, cause it to secrete the pancreatic amylase. Pancreatic amylase will come and it will act on the proteins and convert it into amino acids. We also talked about enterokinase or enteropeptidase that converts the prozymogen inactivate. That means the trypsinogen into active trypsin and chymotrypsinogen into active chymotrypsin. All of those would act on the protein and convert it into amino acid. The lumen of the alimentary canal is made out of protein, then how come we do not digest our own stomach wall or our own intestinal wall? That doesn't happen because our enzymes are secreted as zymogens. They are secreted in the inactive form. That's why every time we have to understand that we are converting them into the active form to act on the protein when there is protein in the body, the gut. Then it acts on the food, and then it converts it into simple. So enzymes initially secreted as an inactive form called as a gynogen or a proenzyme, and pepsinogen is inactive form converted by HCl into, this happened in the stomach. Inactive form converts its HCl into pepsin, breaks down the proteins into polypeptides. Protein digestion, we have done this part. Trypsinogen activated by enterokinase or enteropeptidase. I showed you how trypsin acts on the chymotrypsinogen, converts into chymotrypsin, propeptidase into carboxypeptidase. We have completed all this in the previous slides. Protein digestion in the jejunum by help of the brush border enzymes of jejunum. These are the enzymes, dipeptidases and aminopeptidases in the jejunum that acts on the protein, converting it into amino acids. Now, amino acid digestion and absorption in the small intestine. If you look at the small intestine, we just talked about the sodium potassium pump that helps as a co-transporter also not only to carry the glucose, it also helps to carry the amino acids and comes down to the blood. Also, some part of it take the help of facilitated diffusion and bring it to the blood. So co-transporter and facilitated diffusion, these are the two mechanisms by which sugar and amino acids move down from the lumen of the intestine into the blood. Protein digestion in a nutshell, protein is broken down by pepsin. Pepsin breaks the protein down into smaller peptides by hydrolysis. We have done this. Duodenum, trypsin, chymotrypsin, carbox, we have done this. Jejunum, because of the brush border enzymes, the remaining part of the protein digestion through these enzymes is completed. We have completed the work of sodium in co-transport and facilitated diffusion. So this slide is also completed. So at the end of the entire digestion chemical process, protein is broken down into amino acids in the stomach through pepsin and HCl, then through trypsin, chymotrypsin in the small intestine, duodenum is involved, and then these enzymes in the jejunum through the brush border enzymes, and finally we get amino acid. And then through sodium co-transport and through the facilitated diffusion, we get the absorption done into the blood. Digestion of fat. Fat is triglyceride that forms glycerol and fatty acid at the end of digestion, and pancreas secretes the lipase which will act on the fat and it will break it down into smaller pieces or smaller globules. But why bile is important? Because it does the process of emulsification. Now, what is emulsification? Fat that we get is big chunks. When bile acts on it, bile is secreted by the gallbladder. Okay, so when this is an accessory helping organ, when bile is secreted by gallbladder, it acts on the fat and it breaks it down into smaller fat globules. This process is like a mechanical process of chewing food. Like we did it in the mouth, the same way the gallbladder secretes the bile that emulsifies or breaks the big fat into small fat. This process is called emulsification, and bile does that process. Once they become small fat, then the pancreatic lipase enzyme can work on it and convert it into glycerol and fatty acid. So we have big fat breaking down into small fat. Who does it? Bile. And this process is called emulsification process. Emulsification process. This process, emulsification process. Convert the big fat into small fat. Is it chemical digestion? No, it is not chemical digestion. Then you take the small fat and you cause the pancreatic lipase enzyme to work on it. Now the lipase will work on the small fat and it will convert it into fatty acid and glycerol. That means the digestion is done. This is a chemical digestion. What is bile? Bile is not an enzyme. It is an excretory solution in the liver. It contains bile salts that emulsify the fat. That means they break the big chunks of fat into small chunks of fat, break fat into smaller pieces. Once they make it small pieces, then the pancreatic lipase can work on it and it will break it down into monoglyceride and fatty acid. So, like for example, if you look at the mouth can do chewing, and then comes the amylase from the saliva, and it breaks the carbohydrate into glucose. This is mechanical digestion. This is chemical digestion. Same way, when the bile comes, the bile breaks it big fat into small fat, kind of a mechanical digestion. Then comes the pancreatic lipase, and it breaks the fat into fatty acid and glycerol. This is the real chemical digestion. So bile from liver, emulsification of the fat by the bile salts. So look over here how the emulsification happens. The big fat is broken down into individual small fat droplets because the bile is secreted and it is attaching to these fat pieces and it is forming small pieces. Then comes the lipase from pancreas and acts on these small pieces to break it down into fatty acid and glycerol. This is the mechanical digestion. This is the chemical digestion. I'll just take a few minutes more. We are almost done. Fat digestion in a nutshell. So first pre-treatment is emulsification. Pre-treatment is emulsification to break the big fat into small fat. Okay. And then the real chemical digestion, lipase acting on the small fat, forming glycerol and small fatty acid chains. Okay, chemical digestion and absorption of the lipids. So absorption, once the small fats are formed, fatty acid and glycerol, these bile salts and lecithin will combine with the fatty acid to form micelles. Fat is released by the micelles to diffuse into the epithelial cells. So this is the first step. Bile plus lecithin plus small fat is going to form micelles. These micelles will combine with the protein to form chylomicrons. And then these chylomicrons will enter into the lacteals and then will be transported to systemic circulation. Let me explain to you. So fat, it's a big fat. Okay? It's broken into small fat. How? Because of bile. What is the process called? Emulsification process. Now the small fat is going to be acted upon by pancreatic lipase. Pancreatic lipase is going to break it into fatty acid and glycerol. All the small fatty acid glycerol are going to go and get absorbed into the blood. All the big chains of fatty acid, they are going to be covered with bile and lecithin and form micelles. These micelles are going to get coated with proteins to form chylomicrons. These chylomicrons are going to go get into the villi or the villus of the intestine and get into each individual lacteals. And then from the lacteals, this fat are going to go into the subclavian duct. And that's different, heading into the lymphatic system. So that's how fat is broken down and transported. So here it shows you the villi and the lacteal, the green lacteal. So the fat, bile salts, big fat, broken into small fat through emulsification, and then these small fat droplets, after the action of the emulsification process, they're coated with bile salts. They'll form micelles made of fatty acid, monoglycerides, and bile salts. These micelles are again coated with protein to form chylomicrons. The protein will bind with them to form chylomicrons. These will enter into the lacteals, and from there they will travel by exocytosis. They will enter into the lacteals, and they will be carried into from the intestine into the lymphatic system. So here it shows you step by step, whatever I explained, the emulsification process, the free fatty acid, the bile salts coating them, how the micelles are formed. Each of that is explained step by step over here, whatever I just explained to you. The last part, we'll go to the accessory. We did salivary glands, parotid. I showed you the structure and the location. Sublingual, below the tongue. Submandibular, below the mandible or the jaw. We talked about the pancreas, exocrine and endocrine function. We talked about the bile coming into the digestive system and doing the emulsification. So let's look at the liver. The liver structurally consists of 4 lobes: left, right, caudate, and quadrate. Caudate means the tail part, and the quadrate, and the falciform ligament, it separates the right and the left lobes. We have a hepatic vein that goes from the intestine to the liver, and blood rich in nutrients. So the hepatic artery brings the red oxygenated blood. The hepatic portal vein carries the nutrient from the intestine to the liver. It does not contain oxygen. Okay, so the hepatic portal vein, it comes from the intestine to the liver carrying the food. It does not have oxygen. The hepatic portal artery, it carries the oxygenated blood from the heart. No nutrients. So remember which one has nutrient, which one does not have. If this has nutrient, it does not have oxygen. This one carries the oxygen, does not have the nutrient. And then finally, the inferior vena cava is deoxygenated blood back to the heart. Okay. Now let us look at the structure, histology or the structure. It's very simple. Though it looks complicated, it's very simple. Liver is also called as hepar, H-E-P-A-R. That's why we have the term hepatocytes, hepatic, all of those. So hepatocytes, all the cells of the liver are called hepatocytes or hepatocytes. They have space between the chains of hepatocytes. So first thing, the cells are called hepatocytes. There are big spaces between the cells. These spaces are called as sinusoids. Space between the chains are called sinusoids. And these sinusoids, look at the sinusoids. I don't know if you can see clearly. These are the green dots that you see. These are the sinusoids. And they're lined by simple squamous. They also contain Kupffer cells. These Kupffer cells are important because they have macrophages that will fight against infection, engulf the debris and the damaged RBC. If a person has jaundice or liver cirrhosis, Kupffer cell activities gradually diminishes. They have a main central vein and they have hepatic triads. Three things are there. Sorry. They have a hepatic portal vein, they have a hepatic portal artery, and they have a bile duct. So these 3 form a triad. So blue, green, and red. The blue is the vein, the red is the artery, the bile is shown as green. So triad, we have hepatocytes, spaces in between are the sinusoids, Kupffer cells, which have macrophages to fight against infection. This is the central vein. This is the direction in which how the blood flows from the hepatic portal vein and artery to the sinusoids, the central vein, hepatic veins, inferior vena cava, taking the bad blood back into the heart for filtration. Bile moves through the hepatocytes towards the canaliculi, bile duct, left and right hepatic duct, common bile duct, duodenum. So the bile that is produced in the liver moves in this direction. The blood enters like this and leaves it in this direction. So if you look at the liver, we are having 2 ducts coming. One is the left hepatic duct, one is the right hepatic duct, both coming from the liver. Together they form common hepatic duct. So like this, left and right come and join, and this is the common hepatic duct. Here is the gallbladder. The branch that comes from the gallbladder is called as the cystic duct. So here's the gallbladder. Its branch is called as cystic duct. This is the common hepatic duct. This is the cystic duct. Both of them join together and form the common bile duct over here. So 2 comes from the liver, 1 comes from the gallbladder. Together they join and they form the common bile duct. All of that enters into the duodenum. The common bile duct joins with the pancreatic duct at the ampulla of Vater. So right here at the ampulla of Vater, you have the duct that is coming from the pancreas. Here it joins with this duct that is common bile duct, and together at the ampulla of Vater, they enter into the duodenum. What are the functions of liver? Blood coming from the intestine is filtered here. So liver is basically the chemical factory of the body. All the toxic substances are removed there. Deamination is done. All the ammonia is converted and removed, converted into safely into urea. All the chemicals, toxic substances are cleared up in the liver. So it's called as the chemical factory. The removal of glucose, amino acid, iron, vitamins, removal and degradation of toxins. It cleans up all the toxic garbage that we eat. It secretes albumin, fat transportation, clotting factors, and our cholesterol, LDL, HDL, and all the stuff is connected to the liver. We'll talk about that later on. Then when we talk about about heart, then between meals, it helps in converting it takes the glycogen and stores up in the liver. Glucose is converted into glycogen and storehouse of glycogen is the liver, and circulation of or used by cells of the body. So the multiple functions of the liver is very, very, very important. Okay. So this is the last tough part. After that, things will become easy. So macrophages. So in our spleen is called as the burial ground of the RBC. Our RBC has a lifespan of 120 days. After 120 days, it dies in the spleen. It's buried in the spleen. So it's called as the graveyard of RBC is the spleen. And macrophages in the spleen, they break down the RBC down, like kind of a recycling process. RBC is broken down, so hemoglobin is released. Hemoglobin splits into heme and globin within the macrophages in the spleen. Okay, so now once hemoglobin of the RBC is broken into heme and globin, the globin is used to make amino acids and it's reabsorbed back into the body. We don't have to do anything with globin. What we do is with the heme or the iron part, that is converted into biliverdin. This biliverdin is then converted into bilirubin. Now, bilirubin is an unconjugated and fat-soluble one. Now, this bilirubin binds to the albumin protein, to another albumin protein, and it travels in the blood to the liver. In the liver, once it is sent, the bilirubin albumin is sent into the liver. In the liver, the unconjugated is made now conjugated. What is the meaning of unconjugated and conjugated? This was fat soluble. We're now going to make it water soluble. So it becomes now conjugated by adding glucouronic acid. So you take the unconjugated fat, unconjugated one, the fat soluble one, bilirubin, and convert it into water soluble conjugated by adding glucouronic acid, okay, to the bilirubin. Now, this conjugated bilirubin is secreted into the bile. So you know that liver produces this bile. This bile is containing conjugated bilirubin. Then that is excreted as a component of bile and travels to the intestine. Now, in the intestine, what is its work? Its work is to act on the big fat and make it into small fat, which is called emulsification process. Now, once the emulsification process is done by the bile, we don't need the bile anymore. What are we going to do with it? We take the conjugated bilirubin in the intestine, bacteria breaks down into it, breaks it down into urobilinogen, which gives the brown color into the feces. And some of the derivatives are also secreted via kidneys into the urine. That's the whole story and saga of the birth and the death of bile. Ultimately, bile comes to the splitting of RBC, hemoglobin, in the spleen. So because hemoglobin, our blood, broke into heme and globin, that's how we got heme. Then it was unconjugated. We attach it to a protein, send it into the liver. In the liver, glucouronic acid converts it into conjugated, and then it is secreted into the bile from the liver into the duodenum. In the duodenum, it's going to convert the fat into fatty acid and glycerol. Sorry, it's going to break the big fat into small fat. Once that is done, we don't need bile. It's converted into urobilinogen and then out, gives the brown color to the feces, out of the anus or through the kidney, out. So the start of bile and the end of bile and how it helps in the fat emulsification process is explained here. So the step by step, hemoglobin released from RBC converts into heme plus globin. Let's not think about globin. It goes back into the body. Think about heme. It converts into biliverdin. It's unconjugated. Send it attached to a protein to the liver. The liver is going to convert it into conjugated, put it into the bile, secrete it through the liver, goes to the intestine, does its work of emulsification, waste is urobilinogen out through the feces. Gallbladder, that's just hiding below the liver. It is an intraperitoneal structure. The layers are the same mucosa. It doesn't have submucosa. The muscles are smooth muscle contraction, and the serosa consists of visceral peritoneum. Stores and concentrates the bile that is produced by the liver. It contains bile salts, cholesterol, bile pigments, bilirubin. Cholecystokinin released after a meal causes, so we know that CCK that is released by the duodenum goes to the gallbladder and causes it to secrete bile that can do emulsification in the small intestine. The bile passageways, right and left hepatic ducts. From there, it comes through the common hepatic. The bile from the gallbladder comes through the cystic duct. So, liver, pancreas, gallbladder. Okay, this guy comes through the cystic duct. This guy comes through the common hepatic duct. This one comes through the pancreatic duct. All of them meet at the ampulla of Vater, enter into the duodenum of the intestine. That's the simplest way to remember it. So we have the liver, left hepatic, right hepatic, common hepatic, gallbladder, cystic duct comes. So cystic and common hepatic join together to form common bile duct. Liver, gallbladder, common bile duct. From the pancreas, pancreatic duct meet at the ampulla of Vater. This is the common ampulla Vater, enters into the duodenum. Here's the sphincter of OD, that is a kind of a valve or a sphincter that regulates the flow into the duodenum. The same thing is shown in this picture. Causes gallstones if there is excess amount of cholesterol. Then we come to pancreas. It's quite simple. Two types of cells. Pancreas is a mixed gland. Why it is called mixed? Because it is both exocrine and endocrine. If it is exocrine, it has duct or pipe, and it has serous acinar cells or acinus, and it secretes these digestive enzymes, amylase, lipase, acts on the protein to convert it into amino acids. These are the enzymes, trypsinogen, chymotrypsin. We already studied these and their activities. In the endocrine portion, it has islets of Langerhans that produce insulin and glucagon. We know the work of insulin. It converts the glucose into glycogen. Glucose converted into glycogen. Glucagon hormone is antagonistic in function, opposite in function. What it does, it reverse work. It converts the glycogen back into glucose and releases into the blood. So what insulin does is glucose to glycogen. That is work of insulin. Glucagon hormone is antagonistic, opposite in function. It will convert the glycogen back into glucose and release into the blood. This will take away your sugar and put it into the liver. This hormone will take your glycogen, break it into glucose and release it into the blood. That means glucagon is going to increase your sugar level, whereas insulin is going to remove your sugar level. This is a question in the exam. If you look at the cells under the microscope, islets of Langerhans look this way. 99% of the pancreas consisting of exocrine part, 1% is endocrine. Then what does exocrine pancreas make? So the CCK hormone from duodenum goes and activates the pancreas, so it releases a bulk of enzymes, amylase, lipase, ribonuclease, deoxyribonuclease. These enzymes work on DNA and RNA. Lipase works on fat, amylase works on carbohydrate. These trypsinogen, procarboxy, all of these are enzymes or gymosins. They act on proteins, and we know who activates them. Enterokinase that is present, or enteropeptidase, it converts trypsinogen into active trypsin. After that, trypsin takes over the role and it converts all of this inactive into activated. And all of this work on the protein to convert it into amino acid. Bicarbonate, it neutralizes acids in the duodenum because thanks to secretin. So if there is a liver dysfunction, then you'd have hepatitis, cirrhosis, all of these diseases, and pancreatic cancer. Where does food go once it is absorbed? So once it is absorbed, once digestion is complete, the food monosaccharides and amino acids will go through the hepatic portal vein. All the nutrients will go to the liver. Liver will extract the nutrient from them. The short fatty acid goes to the hepatic portal system. The long fatty acid, as I explained earlier, will enter into the villus lacteal and go into the left subclavian vein. So with that, it finishes off